Am I audible? Good morning. Morning. So we are here to discuss about science and technology for upcoming prelims, right? And in next 3 days, including today, we are going to cover some key issues, right? I mean, 3 days are not too much to cover it in entirety, right? But still we can just brush up through the entire concepts and some important area. So today we are going to discuss about two topics. We'll take it, right? Because every day we are going to cover something. Like we know that in prelims examination they ask question, right? So if we talk about prelims, so the syllabus says it's general science, right? So in syllabus, अच्छा ठीक है, no issues. Right. So prelims 2024, and we are here to discuss about science and technology. And as per UPSC syllabus, it is called general science, right? But if you look at the question paper, then things are quite different. Normally, I say that general science, what we perceive it like biology, physics, and chemistry. So that is what we call it general science. But then the question which are coming in UPSC examination are not coming from this area, right? You hardly find any question from chemistry. In last 10 years, you will find 1 or 2 questions from chemistry. And that is not chemistry even, that is applied chemistry, right? Then you will hardly find any question from physics. In last 10 years, they might have asked 3 to 4 questions in physics. In biology, you will find some question, and after COVID, immunity, virus, vaccines, that become the important area. So it has also got some kind of background, right? So biology, physics, chemistry, it's all fine. It's good for basic concept, but the questions are not coming from there. UPSC is asking invariably around 12 to 15 questions, right? So 12 to 15 questions are coming from general science area. But what general science is exactly? It's main topics, mains topic. So mains topic is, let's say, biotech, questions are coming from there. Nanotech, questions are coming from there. Then space tech, questions are coming from there. Information technology and computers, you can see questions from there. So these are the area which has been mentioned in mains coverage, right? I'm not considering robotics because robotics is more or less like mains topic, not for the prelims. But biotech, nanotech, space tech, IT and computers, these are the chapters which has been mentioned into the main syllabus, and questions are majorly coming from this area. You can add few more things like, let's say, defense technology, right, and nuclear technology. So these two technology has not been mentioned in science and tech syllabus, nowhere, but you can see questions are coming from these areas, right? So this is something which is being suggested by UPSC without explaining that what they're going to ask. That is why we always say in UPSC examination, it's always said that you will get to know about the syllabus only when you look into the question paper, past year question paper. I mean, nothing makes sense except you look into the past year question paper. And once you look into the past year question paper, you will find these areas are very important, right? So let's forget this, because this is a time when we have to focus on those areas from where the questions can come in. Although basic biology, basic physics, basic chemistry, that is required, and that is why we start our preparation with NCERT, right? So when you read NCERT 9th and 10th, you get enough knowledge about the basic concepts of all these 3 areas, right? And it doesn't mean that NCERT reading means that you need to read each and everything into the NCERT. It's just like, some concepts, principles, theory, examples, which is, they're in our daily life, right? Not like that we have studied in 9th and 10th class. No numerical, no formula, no equation, no balancing of equation, no calculation, just text and its connection with daily life. That's it. But here, it's very important. Mains syllabus, 4 chapters and 2 additional chapters. Today, we'll try to cover these 2 areas, biotech and nanotech, some important aspect, because we have to cover the current development as well as fundamentals. One more thing, that kind of question the UPSC ask. For that, you need to look into the question. Sometimes we consider that current affair is the only thing that we should cover. But if you carefully look into the question paper, they are not asking any question from news, right? So current affair has got 2 things, right? Current affair has got like one news element, the face value of the current affair. Another is the background, the fundamentals. I'll give you one example, right? In 2020-21, in Union Budget of India, there was something called NMQTA, National Mission on Quantum Technology and Application. National Mission on Quantum Technology and Application. After that, you know that India became 7th country to start NQM, National Quantum Mission, right? So things started in 2020-21 when National Mission on Quantum Technology and Application was rolled out, and there was money has been allocated. We'll talk about that. And after that, National Quantum Mission was initiated by India with one of the target to develop quantum computer in next 5 years to 8 years time span, right? So this is what? This is news. But what they asked, UPSC asked simple question, and the UPSC asked question that what is qubit? You get my point? What is qubit? So now, no matter how deeply you go into NMQTA and NQM, you will never get to know about qubit unless you talk about the fundamentals of quantum computers. You get my point? So this NMQTA, let's say, or NQM, has given some of the keywords like quantum computer, quantum sensing and quantum metrology, quantum encryption, quantum communication, and then there is quantum material and quantum device. Now we have to explore all these topics. You got my point? It's just a keyword which has been given, because in NQM, government is saying that we are going to talk about 4 verticals in quantum technology. One is quantum computer, another is quantum communication, quantum encryption, another is quantum metrology and sensing, quantum device and material. So we need to get into that. And then they ask one question of quantum computer, that what is qubit? Like we, in digital computer, we have bits, here we have qubits. But then it's the start of the thing, right? First time UPSC asked any question from quantum computer or quantum related question, which is directly relating with the quantum computer, right? But that's a start. Now lots many things would come up. Now if you, obviously this is important news, we have to talk about this because some question can come directly from this also. But if you look into the 12 question, you will find out of 12 question, only 1 or 2 questions are being asked from plans and policy, by naming plans and policy. 10 questions are coming from where? Fundamental knowledge, context of the current. The current has got 2 things. So if we talk about current affairs, that's the biggest thing in UPSC preparation, right? So from my view, current has got 2 elements. One is news and another is context of that. And context means the fundamental part, right? Fundamentals, right? News has memory and recall value. You know it. If you are reading, you will get it. And that is why if you look into the entire material market of UPSC, you will always find now they have also changed the way. They are mentioning a news, and then after the news, they are also explaining the fundamentals of it, right? You got my point? So for that, you have to take some clue. Anyway, So from my point of view, that is what we have to follow. Fundamentals are very important. News are indicative. They indicate and they say that, okay, this things has been important. Let's say in biotechnology, if we talk about DMH11 is very important right now because lots of things are happening in the Supreme Court. Case and PIL has been filed. Government has to say something else. Environmentalist has to say something else, right? And court is trying to create a balance. So there is a lot of dialogue going on. And then we need to understand that what stand should India have on GM crop. And then example is DMH11. Now, DMH11, I mean, that's a news, but then concepts are there, right? So we'll go with that one by one. So we'll start with first topic we are going to take, that we are going to talk about some concepts of biotechnology. Now, in biotechnology, the first concept that we are going to discuss is genomics, right? And you all know genomics is nothing but the study of genome, right? And genome is what? The total sum of genes in an organism, right? So that is what genomics is. It's a very fundamental thing in biotechnology, and lots many things started coming in this area. And to study genomics, we have many programs that we have rolled out. Let's say at world level, if we divide it into this, then at the world level we have Human Genome Project, right? That is called HGP, and it is called HGP Read. Then the second we have HGP Write, which is already going on, right? Then the third one is called Earth Biogenome Project. Earth Biogenome Project. Now we'll try to understand the difference between all three. So genomics is a study of genome. I mean, we just want to study the genetic makeup of the human being, right? When it comes to human, it's a human genome project. And the prime motive behind studying the entire genomic structure of the human being is to find out the possibility of disease, the background of disease, the genetic basis of disease, to find out the way to treat the disease, right? Because everything is written in our genome. Our genetic makeup is everything. It's a blueprint of our life. So unless we understand the entire mechanism and the life process, we won't be able to create some kind of breakthrough in terms of medicine, in terms of better health, in terms of better, life. So for that, we started genomic study, right? The first genomic study that is started in 1990 was Human Genome Project, and now it is called Human Genome Project Read, because there what we did, we have read all the genetic information present on the human body, right? Like we have DNA, and DNA has got adenine, guanine, cytosine, thymine, that sequence. The sequence has been deciphered, and this all information was, transferred to a supercomputer. Now supercomputer can analyze and it can give a lot of information about it, right? So this Human Genome Project was an international thing initiated by USA. Then all three things are basically international effort. In Human Genome Project Read, we took the DNA out of the body, we read all the information, and then we transferred those information into a computer system so that we can analyze, we can find out some traits of the human genome, and on the basis of that, we could find the genetic basis of some disease, some genetic disease, some non-genetic disease. And then once we understand the genetic basis of disease, we would be able to find the better medicine out of it, right? The main purpose was that only, to improve the quality of life, right? One thing you should be very clear, that technology has the ultimate purpose, and the ultimate purpose is to improve the quality of life of the human being. Now we have to connect the means and the way to it. After completion of Human Genome Project Read, where we have to read the DNA, then we went to the Human Genome Project Write. And in Human Genome Project Write, the things was done in a reverse way. Here what we have done, we have studied the DNA and transferred the information to computer. Now Human Genome Project Write, first we created a information base on computer and then we transferred it to DNA. You get my point? The Human Genome Project Read was taking the human DNA out, reading it, putting the information in computer. In Human Genome Project Write was creating the information structure on a computer, and then on the basis of that information structure, we have created a DNA molecule in laboratory. It means that you have already created one structural data, and then this data will be transferred to the DNA in laboratory. So this is basically here what we have done. Here we have done Human Genome Project Read, that synthetic human genome has been created. Right. What we have done in Human Genome Project Read, so here what we have done, that genetic information has been studied. So it's just reverse. And these both are for human being. The information you have read, that is Human Genome Project Read. Information that you have written, that is Human Genome Project Write. Purpose is both. Purpose is to understand the human genome better, once by reading it and another by creating it in our own way. Now we have done this, we are going through this. Next project was that why can't we broaden the horizon? If genomic study can reveal us so much of important data, then why only human being? There is a lot of organism around us that we consider as resources to human being, like crops, animals, insects. These all are biological resources to us. I mean, we source our crops from the around only. We have animals around us. We have many microorganisms that we are using right now. We are always looking for alternative nutritional avenue, right? We are always looking for biofuels, and for biofuels we need to get some good microorganism which can do fermentation, right? So for that also we need to explore all the living creature around us. So Earth Biogenome Project is that genomic study of every possible organism present around us, around 15 lakh of them, plants, animals, and protozoa, all of them, right? So here what we are going to do, Earth Biogenome Project, genomic study of all eukaryotic organism, like plants plus animals plus protozoa. So all plants around us, all animals around us, protozoa. Eukaryotic word you must be knowing. The cell, a kind of cell which has got definite nucleus. If we consider the entire biology into two parts, eukaryotic and prokaryotic, then bacteria, blue-green algae, these two parts are prokaryotes. Rest of the other things are what? Eukaryote. So in this Earth Biogenome Project, we are not going to talk about bacteria, blue-green algae, and mycoplasma. So except I can write it so that conceptually you can have it. If we write it, then except prokaryotes. And what are the example? Bacteria, blue-green algae, and mycoplasma. I mean, at this point of time, if you remember, these three are prokaryotes, it's all fine. So except for all these three, we are going to study all of them, right? So that is what we are doing in genomics. This is also going on. It's already in process. India is participant in this Earth Biogenome project, right? But what India is doing in that? Obviously, India has to do something in genomics too. India has already taken some step in genomics because genomic data means human genome data, and this data will reveal a lot of information about our health status, disease, the way to fight with disease. But then it is very important to have our own representation, right? Every continent, or let's say every geographical location, climatic locations, have different set of climatic conditions, and according to those climatic conditions, different types of disease would appear, right? Let's say if you go to the Europe and America, there you will find skin cancer is the most common cancer, right? If you come to India subcontinent, then you will find lung cancer, throat cancer are the most common cancer, one of them. You get my point? We are not bothered about skin cancer. So their data cannot be very useful for us, right? Because if America is doing human genome project, then obviously American population has represented it. Their people from Indian subcontinent and their representation is less. They have their own genetics. We have our own genetics. At the time of COVID, it became very evident. People started talking about the whole thing, like Europe and America, they are not able to fight with COVID the way people of Indian subcontinent can fight. You get my point? It means that there is a little bit difference between our genetics and their genetics. And it happens, like every ethnic group, every geographical isolation, there would be some kind of a little bit of genetic differences. But this little bit genetic differences can play very important role. So initially, we accessed the data of Human Genome Project, and then we started studying on that in India so that we can find out some root cause of disease which is prevailing in India. Like lung cancer, throat cancer are the major things which are happening in India. India is almost turned into diabetes capital, right? So there are certain set of disease that is affecting Indian population, right? And those set of diseases are not prominent in Europe and America. They have their own problem. So we have to study our own people's genome, right? Although in Human Genome Project, 1,000 people's presentation was there. Out of 1,000, majority of the persons from their European and American descent. Very few were from Indian subcontinent. So we wanted to do it in some other way. So then what we have done, Indian efforts. So India in genomics. So India in genomics, right? That is the important one. So we have done two things. One, we are doing genomics in agriculture. Agriculture. And agriculture for what? Crop improvement. Crop improvement. That we are doing. So even we study the genome of the crop and we can improve it, right? We have done genomics, genomic study of rice, wheat, right, gram, then turmeric, haldi, tulsi, ragi. We have done a lot. Lots many things we have done. And the purpose of this is to improve the crop variety, to get the better crop variety. But the biggest thing that we are doing is in health sector. Health sector. Right. And in health sector, we need to study the human genome. And for the human genome, what we are doing here, we have started something called GIP, Genome India Project. So Genome India Project is nothing but the genomics. And in this genomics, Indian representation is there. People from Indian Indian descent, that will be, taken as sample, and then we are doing it. So, Genome or genomic study of Indian population. Right. That is called GIP, Genome India project. That's the biggest one, it's going on. Right. So 10,000, Indian national, that would be taken in, and we'll study the genomic data of that. So here 10,000 Indian genome will be studied. Why we are going to study it? So that we can get the genetic basis of disease which are prevailing in India. That's it. Whenever they talk about genomics, the first thing about human genomics is disease, relate with the disease, right? So that is what we are doing. And the second thing that we have done, that you must be knowing, that is called INSA-COG. INSA-COG. It's not for human being, it is for COVID management, right? You know that COVID has changed everything around us. Even the UPSC is taking a lot of things from COVID, right? So INSA-COG is Indian SARS-CoV-2 Consortium for Genomics. Because you know that COVID has got a different, first Delta variant came, created a lot of problem, then Omicron came. Every time a new variant would come up. Every year 2-3 new variant would make news. But I'll tell you, these are not only 2-3 variants which are making news. We just know those variants which are variant of concern. WHO created like variants of concern. So variant of concern means this variant has got something which can impact on our life and health. But there are many variants which come up, and these are not variant of concern, so we don't talk about it. We simply don't talk about it. Because if viruses are there, COVID is virus obviously. So virus, if some variation is coming in virus, so first of all we need to understand that how the variation would come in virus. So you know that a virus is nothing. A virus is like this. It's a simple structure. It's like protein capsule, and in protein capsule you have DNA or RNA, right? Or let's say if we talk about COVID, this is like this. There will be a spike protein, right? It's a protein structure. It's entirely protein, and then inside you will have DNA or RNA. This is virus. Virus has no cell. So this protein structure would not have any variation. It's like almost same. And this has got DNA or RNA. If I talk about COVID virus, coronavirus, it has got RNA, right? So this is RNA, and you know that DNA and RNA are genetic material which has got code on it. According to that code only we have different kind of structures like I have, you have. So virus also get its character from its DNA or RNA. If there is a slight change in DNA and RNA of virus, then it's a new variant. And if new variant is coming up, then it could be harmful, it could be less harmful. Sometime there are certain change in RNA of coronavirus, and that change has decreased the potential of the virus to impact on our health. So we don't bother about that variant because the variant of non-concern, it's not a concern. Because the virus has gone weak, it is not impacting our life. It won't be creating any problem. We'll get infection like every kind of flu or influenza kind of thing, but it won't be life threatening. But sometime variation could come up in a other way. Some sudden change in RNA, and then the virus has become more stronger. Its mortality has increased. Infection rate has increased. And then it is called what? Variant of concern. Now the virus do not have any kind of facial thing, right? If I come tomorrow with a new haircut, you will understand, okay, sir has got some change in looks. But virus you cannot see. And even if you see the virus, you won't be able to see the variation just by looking into it. Because variations in viruses are coming in RNA or DNA present inside. And RNA, DNA has got nothing but adenine, guanine, cytosine, thymine. If it is RNA, then uracil would be there. right? So if you want to study a variant of a particular virus, then you have to study its DNA and RNA. And the moment you study DNA and RNA, it becomes what? Genomics. You get my point? That is why long back when this COVID was happening in India, then it was like gene sequencing can reveal the new variant of the COVID. And gene sequencing is nothing but the genomics. Gene sequencing meant that sequence in the gene, adenine, guanine, cytosine, thymine, right? So that is what. INSA-CoG is nothing. INSA-CoG is India SARS-CoV-2 Consortium of Genomics. And this is COVID virus. And what is the purpose of this COVID virus? I mean, this consortium. So here, to find out new variants of coronavirus by gene sequencing. By gene sequencing. So if we talk about health issues, then two important aspects are there. One is GIP, Genome India project, where we are studying 10,000 Indian genome to find out genetic basis of different disease and the way to tackle it. And then today we are having INSA-CoG. That's INSA-CoG is chain of labs. It is, chain of labs. And they do what? These labs are doing what? Gene sequencing of what? Coronavirus, to find out the new variant of it. You got my point? So genomics, long back, still it is very important because genomics has become a tool to find out new variants of anything. You got my point? Like every day in every city, in every state, there are some COVID tests going on. If tests are turning into positive, let's say 500 people tested positive of COVID, then 10%, 10 to 20% of those tests would be randomly selected and it will send to INSA-CoG lab. In INSA-CoG lab, they will do gene sequencing of it. They will read the information written onto the RNA of the coronavirus and they will match it with the previous one. If it is matching with the previous one, it's the same variant which has been there in Delhi or anywhere. If there is a mismatch, then it's a new variant. Now this entire data will be sent to lab and scientists would study and they will come up that this new variation, the new code which has led to generation of new variant, how it is going to impact on our life. If these variants are not creating any problem, rather it has become weak, then no problem. That virus has become weak, no problem. This variant will automatically go off. But if the variant is having more problem, then we'll spread this news. And then we always take some, precautionary measure. We'll test it that whether these variants can be countered by the vaccines that we have taken, the antibody in our body that has been, created. If not, then what should be the way? So that we are always at danger of this, right? So this is about the genomic study. I mean, in a nutshell, I tried to complete it. But now with genomics, there has been some new terms, current affairs terms. In my regular science and technology classes, I take current affairs along with the class only. But here I am just taking some important aspect and then I am linking it with the current affair. Some related terms are there in terms of genomics or human genome. The first term is called, nowadays it is called dark genome. What it is called? Dark genome. Right. So, dark genome is what? I'll tell you. Normally we say that it's already been said that in our cell, around 75% genes, DNAs, are not active. They're called junk DNA. What do we call it? Junk DNA. In our body, in our cell, let's say if a human cell is there, it's a human cell. Human cell has got 30,000 genes, roughly 30,000 genes, you can say. But these 30,000 genes are just a small part of it. If we consider all DNA present in our body, then more than 75% DNA are basically junk DNA. They don't have any role in our body. It's just present in that, right? These 30,000 genes are active one, and these 30,000 genes are responsible for everything that is happening in our body, everything that happen in our body. Like our eye color is black, brown, blue, because of some of the genes present in. The kind of skin color we have got, that is due to the some of the genes present in. The kind of metabolic activity going on in our body, enzymes and hormones are being secreted. We are able to digest the food, right? Just because there are certain genes present in those 30,000 genes which are doing their job, right? So 30,000 genes are active part of the DNA. But the major part of the DNA are non-active. And that is why it is called junk DNA. What it is called? Junk DNA. And scientists were convinced that junk DNA do not have any role in our body. It's just present because it's an evolutionary load coming from the background, right? But now scientists have started linking this junk DNA with certain disorder of the body. And the disorder of the bodies are mental disorders, like schizophrenia, bipolar disorders, right? So this is a new connection which is coming up. And they are saying the dark genome are the inactive part of the genome, that is junk DNA. So it is inactive part of genome or junk DNA. Junk DNA, right? It has no active role in protein synthesis in body. You know protein synthesis? Any gene will express itself by protein synthesis, right? If genes are active or not, how can we recognize? If genes are engaged in protein synthesis, it's active, otherwise it's not. Now, why this all of a sudden protein become important? Because our body is made up of protein. And all the functions of our body is being guided by protein, right? So body entirely is made up of what? Protein. Protein is a major constituent of body structure. Anybody is going to gym and trying to build up muscle, what kind of supplement you take? Protein. Because our muscles are made up of actin and myosin protein. Our skin is made up of what? Keratin protein. Hair and nails are made up of keratin protein. Our bones are made up of osine protein. Our blood plasma has got a lot of protein. Albumin, fibrin, globulin, right? So if I say that each and every body structures are made up of protein, then it's true. Yes, along with the protein, there would be certain more thing. Let's say if we talk about bone. So bone is calcium, phosphorus, and then there is a protein called osine. You got my point? But protein would be there. And if the structure of protein is changing, it means the body structure is also changing, right? Let's say if somebody is very fair, then there is a less melanin protein. Somebody is having dark complexion, more melanin protein. It's again protein. You get my point? If somebody is having straight hair, it's alpha keratin. It's a protein. If somebody is having curly hair, it's beta keratin. It's again what? Protein. A little bit change in the protein, your hair texture, hair color, hair structure, that would change, right? And who will decide that which kind of protein should body make? It's gene. Because which kind of protein the body will make, the information for that protein is written onto the genes or DNA. You got my point? So if a gene is active or not, how can we find out? We can find it out with the help of looking into the protein synthesis. If protein synthesis is happening, then gene is active. if there is a certain part of DNA not engaged in protein synthesis, it means it's inactive, right? That is why it is. And in basic biology, we always talk about something like DNA to RNA and RNA to protein. That is how it functions. So DNA is your gene. So this is very important. DNA to RNA, RNA to protein. Ultimate thing is what? Protein. Our body is made up of what? Protein. The structural part of body is protein. Now the functional part, if we talk about, then you know that in our body we have different types of enzyme, and all enzyme in our body is made up of protein. Every single enzyme that our body secrete for any purpose, not only digestion, then it's protein again, right? So if our body is secreting, let's say 500 types of enzyme for different function, all 500 enzymes are proteins. The structure is protein, function is protein, right? We have hormones also, and except for few hormones, all the hormones are what? Protein again. Insulin hormone protein, growth hormone protein. You get my point? So dark genome means that part of genome which are inactive, they do not participate in protein synthesis, they do not have active function in our body. But in recent studies, now they are linking dark genome with some psychological disorders. Now this is very unique. It's a new realm which has been opened. Because till now we had understanding that psychological disorders are basically due to some impact of the environment, some kind of learning, some kind of, unknown factors. But now schizophrenia, that is called dual personality, bipolar disorder, heavy mood swings, that has been linked with this dark genome. So you can write 1 point. इसी के आगे dark genome is now being linked with psychological disorders like bipolar disorder and schizophrenia. Right? So if they talk about dark genome, then you should connect with the junk DNA, no role in protein synthesis, and mental disorder possibility linked with it. Right? Always try to catch the keywords. Right? Because if you are in the last leg of the preparation, then I would expect that you have all the background with you, and then connectivity need to be done. Right? You got my point? Now, along with that, a new term came recently, and that is called eDNA. eDNA. Because we are talking about genomics and related area. eDNA is called environmental DNA. Very unique concept, and environmental DNA. Right? Now, what is environmental DNA? It's very easy to understand. Environmental DNA means that DNA content present in environment that has been coming from any kind of living organism, right? So we have DNA in our own body. Each and every cell of our body is having DNA, right? But then sometime from our body, some cells would come out. Let's say saliva. So saliva doesn't have cell, but obviously mouth lining would break off some cells. So if saliva is there, then saliva would have some mouth lining cells, and out of this saliva we can trace back the cells, and the cell will have DNA. So now that is part of environmental DNA, right? The DNA which is present in environment, but that it doesn't apply on human being, right? Environmental DNA is all about biodiversity monitoring. You get my point? So you can link it with the environment. जहां पे भी junction होगा, that will area become more important. Let's say if a migratory bird is coming from Siberia to India, right? And you know that in the month of November, December, January, February, all over India, people are, monitoring these migratory birds which are coming in and going back, because that's indication of healthy ecosystem, right? That your ecosystem is good enough, that birds are still considering it as a summer habitat or let's say winter habitat, right? But sometimes the birds are very small, sometimes the number of birds are very less, sometimes it is beyond your, visual range. You get my point? But still they get to know about it, because they know that these are the area where the bird would come. If the bird has came from Siberia and stayed over there, it may be possible that human being will miss this bird. They won't be able to record it. They won't be able to click it in a camera. But then they go and they take the sample of the soil and the other thing around it. Because if the bird has came, the feather would come out. The excretory waste will come out. The egg will hatch. Some bird will be dying also. You get my point? And that will release what? DNA. DNA will come from the feathers, because feather ka last mein blood laga hota hai. DNA will come from excretory waste, because they are having food and they are eliminating excretory waste there. Some bird would be dying, their body parts would be there, bones and other part would be there. And they will take the sample, and in this sample they will try to understand the DNA content. And if they found the DNA content of that particular bird, it means the bird has visited, right? So if you want to study the biodiversity composition of a particular area, then you can simply go and put a camera trap and lots of things around it to find, and you can study it. But the other way, that look around, collect the DNA sample from the environment, soil, water, and whatever possibilities there, and then you will get to know about it. So that is the concept of environmental DNA. You can write. It's a content of DNA. It's a content of DNA released by animals and birds. Animals and birds in environment. In the form of excretory waste, saliva, hair. Hair will not have DNA, but hair is pulled, it will have DNA. Hair, feather, dead body, etc. This can be helpful in monitoring of biodiversity, and to identify the migration route, right? So how animals are migrating? What is the route of their migration, right? So this all can be find out from there. That is environmental DNA. Just you need to understand the concept, you will get it, right? Now there is another term that is called CFDNA. CFDNA is cell-free DNA. This year only, the last year only the concept came. And this concept with human being, right? So normally we know that in our body, let's say if we talk about human body, then in human cell, it's a human cell or animal cell, we have DNA at two places, right? This is called nuclear DNA and this is called mitochondrial DNA. What we call it? Mitochondrial DNA. So in our cell, we have two places where DNA can be present, nucleus and mitochondria, right? If I talk about in terms of numerical, then 30,000 genes, that is for indication and 37G. That's the difference, 30,000. That is why we always talk about nuclear DNA. We never specify mitochondrial DNA unless we have some purpose. Because 30,000, that means major things are being, determined by this nuclear DNA only. But we do have another place where we have got DNA in our cell, that is called mitochondrial DNA or mDNA. These both will be known as cell DNA. These are what? Cell DNA. Cellular DNA we can call it. So together we call it what? Together we can call it cellular DNA. But now let's get to the concept. Let's say in our own body only, if a cell is dying, if the cell is dead, and once the cell is dead, the cell will fragmentize. It's very common that in our body, cells will keep on dying, right? And we will survive because every cell has got a definite lifespan. Let's say RBC has got lifespan of 120 days. WBC don't have any proper lifespan, but they keep on dying because they are fighting with the infection agents and the war is ongoing in our body and they will die, right? So muscle cell will also degrade and die. So in our body, cells will keep on dying, new cells will keep on replacing them. But if the cell is dead, then what will happen? Cell is dead, cell will fragmentized and their DNA would come out. Now this DNA will come out of the cell. Because once the cell is dead, the cell is fragmentized. DNA is out. And when the DNA is out, where all you can find the DNA? In our body fluid, like in our blood, in our lymph. So DNA particles, DNA fragments which are present in our blood, plasma, lymph, these all are called cell-free DNA. The DNA which got free from cell. And how it would be possible? Once the cell is dead. You get my point? That is called cell-free DNA, right? But why this cell-free DNA becomes so important? In our body, we have cell-free DNA. It's all fine, right? So it becomes important because now we can get the DNA from our blood plasma also, right? I mean, it's not necessary that we need to get hold of the cell to get the DNA out. Now we can get the DNA from blood plasma. Anywhere in our body, blood is reaching. And if the cell is dying in anywhere in our body, then fragment of the cell and the fragment of the DNA will come into the blood. So even if I take blood from this part, it will have the fragments of DNA of any other body part, isn't it? Now let's connect with that. Let's say if somebody is having tumor, cancerous tumor, right? Cancer, you mean to say cancer. So what is the way to recognize cancer? Biopsy. First of all, you need to get to the tumor. And the moment you realize that your body has got tumor or somebody's body has got tumor, by that time only the cancer has already spread in the body. Because if the tumor is localized, you are feeling that there is a tumor. It means that cancer has already advanced. Cancer should be tracked at the primary stage. When there is a very small tumor in the body, a small tumor means 1,000, 2,000, 5,000 cells tumor. But that is basically a kind of mole, a kind of wart that appear into the body. We don't care about it, right? Because majority of the moles and warts are non-cancerous. But some of them can be cancerous. If it's increasing in size and then we have to visit the doctor and the doctor says that now we'll have to test it by biopsy. Cancerous growth is basically uncontrolled cell division. And the cell is dividing like anything. And out of this tumor, some cells will be dying also. And if some cells are dying, then it will be fragmentized and their DNA would come into our blood. So if I take the blood sample from anywhere in the body, we can find the DNA of cancerous cell as well, right? Now, DNA of cancerous cell, is it true? Yes, it is true because cancer in most cases is because of change in our genetics. That is called mutation, right? There are two types of cancer. Nowadays, you know that because every news that makes sense, that should make sense. अभी इंडिया में क्या चल रहा है सर्वाइकल कैंसर को लेके कितनी सारी अवेयरनेस चला रहे हैं तो देखो सर्वाइकल कैंसर, राइट, यू गॉट दैट, एंड फीमेल आर प्रोन टू इट एंड दे मस्ट बी एडुकेटेड फॉर दैट, दैट इज अनदर थिंग. बट सर्वाइकल कैंसर होता किससे? देयर इज अ वायरस कॉल्ड एचपीवी, ह्यूमन पैपिलोमा वायरस. अब क्वेश्चन ये थोड़ा ना आएगा सर्वाइकल कैंसर किससे होता है. क्वेश्चन क्या आएगा कि इन ह्यूमन बीइंग ट्यूमरस ग्रोथ कैन लीड टू कैंसर. According to your view, which of the following can be reason behind cancer, causative agent of the cancer? Consider the following statement: mutation, radiation, pollution, viral infection. अब बताओ इसमें से कौन कौन. So at that time you might miss this viral infection, but again you know that that cervical cancer is because of HPV virus. So all four will be there. You get my point? So that is how we need to connect. It's not like face value they are going to ask. If they ask that cervical cancer is due to what, तो ठीक है ना, बनाओगे. So easy. But do you think that if cervical cancer is due to HPV virus, so is this question is going to create any differentiation? ये तो सब बना के आएंगे. You get my point? So don't talk about those questions, those 10-20 questions that everybody will do it. क्योंकि that is non-differentiator question. It won't create any difference. उसकी तो बात ही नहीं करनी. We have to link in some other way differentiator question. Last 20 question that we attempt, that is the differentiator one, right? So I mean, it could be like two-way. Cancer can be triggered in our body in two ways. One is by viral infection. Now we know that there is one example of HPV, human papilloma virus, right? And the second one is, and not only that, human papilloma virus is creating cervical cancer, and then there are certain viral infections that can lead to oropharyngeal cancer, that is cancer in growth, and that is also due to virus. So viral infection can lead to cancer, but not all. There are certain viruses which can lead to cancer. And the second is change in our genetics. That is called mutation. What we call it? Mutation. Now mutation can have different causes. Mutation can be caused by radiation. Mutation can be caused by pollution, like pesticide pollution, like pollution in the food, pollution in the environment, pollution into the water. You get my point? But then, if there is lung cancer, if there is, other kind of cancer, then it's a mutation. It's a sudden change in genetic material, sudden change in DNA. So DNA has changed. And if we take the DNA out of the blood, and then we see those DNA changes out there, then we can relate with the cancer. That is why cfDNA has turned into the way by which we can give our blood to the lab, and the lab will test that whether in our body we have cancer or not. So easier way to detect the cancer. You got my point? That is the one way. Because if there is a tumor means uncontrolled growth, cells will be dying, coming out, fragmentized, DNA will come out, DNA will float in blood. We take the blood, we'll do it. You got my point? That is one way. The second one, let's say if a woman is pregnant and she's having a fetus inside her womb, and we need to test the fetus genetic makeup, because it has been suggested by doctor that the fetus or embryo might have some genetic disorder, and the baby born out of this pregnancy will not be normal. So Indian government allow that if there are medical conditions and the baby is not able to live a normal life, then parents and doctor can agree upon, and then the baby can be aborted within 24 weeks of the pregnancy. Isn't it? But for that you need to test it. You cannot take it like 24 weeks ke pehle aise hi. So you know that testing is done by something called, earlier technology is, that is called amniocentesis. With a long syringe, you just pierce through the belly of the mother, and then you try to catch some fluid from around the embryo, because embryo in mother's body is in bag like or sac like a structure. Embryo udhar hi rehta hai, and there is always a liquid present in it. So we all were in mother's womb in a floating condition. Now baby's body will also break off some cells. हमारे body से भी cells निकलता है ना, skin cells always comes down. So baby's body will also release some cells. And these cells will be present in what? In this amniotic fluid, in this mother's womb fluid. This fluid will be taken out and then it will be analyzed. Cells will be extracted, analyzed. But then it's a dangerous thing because a little bit of mistake and that may lead to miscarriage because you're interfering with the very personal space of the baby. I mean, personal space means that space, it's a biologically separated one. Biology never says that you interfere with that. We are interfering because of the technology and because we know that there are certain things which cannot go in the favor of the baby. So that is aminosynthesis and it is something that we do it right now, but it's not advisable. But one thing we know that baby is connected to mother by placenta and umbilical cord. So baby and mother are connected. You get my point? And now it is being said that baby's cell which is coming out, right? That will fragmentize, DNA would come out and that DNA particles will enter into the mother's bloodstream. Where it will go? Mother's bloodstream. And if you take the mother's blood out, then still you can get the DNA of baby. And now by testing the mother's blood, you can find out whether the baby is having some genetic disorders and problems or not. You get my point? That is called non-invasive prenatal testing. Non-invasive because there is no invasion with the baby's own system. The moment you pierce through this amniotic bag and get the fluid out of the baby's surrounding, then it's invasive one. Invasive is not something which should be practiced. Non-invasive one, what? Take the blood of the mother. Anyway, if the female is pregnant, every 2-3 months some testing need to be done, right? So that is another way. मैं अभी बता दूंगा अभी इसको. Third aspect of it, because this CF was, Hindu newspaper में पूरा एक page था, big, but there were 3 main issues that we are focusing on. Third is that let's say if there is a patient and the patient has taken kidney transplant, right? So kidney has been transplanted into the patient. Now you know that if kidney has been transplanted into the patient, matching of the kidney is done, right? Proper match should be there. But nobody in this world can match 100%. You know that? Nobody in this world will match 100% with my body. And my body has just one rule. Everyone's body has just one rule. Self and non-self. WBC, they recognize self and then after self everything is non-self. You got my point? And WBC will always try to kill and destroy everything which is coming from the other side. Right? But still, doctor will do a lot of calculation. Doctor will say that okay, we'll find out the suitable match, and once they get the suitable match, they will transplant the kidney. But still, WBC will not spare that kidney too. WBC will consider that kidney which has been transplanted as a foreign body into the own system, and WBC will wage a war against it. Right? That is why after every transplant, the patient are put on immunosuppressant medicine. Their immunity system are suppressed so that WBC do not become overactive to eliminate and kill the kidney which has been transplanted. You got my point? And for that matter only, we keep the patient for 15 days, 1 month, 2 months on that medicine. Immunity systems are purposely suppressed. That's a very dangerous situation because the immunity system is suppressed. And when immunity system is suppressed, then you are prone to many infections, right? So in a sterilized environment, they have to keep it. But still, there are certain chances that kidney can be rejected. Many times it happened, right? So whether the kidney is accepted or rejected, we can get it from CFDN. Because if the kidney is being rejected, you get my point? Then WBC will attacking onto the kidney, killing the kidney cell will be decomposed, fragmentized, DNA will come out and DNA will be present in the patient's blood. So they keep on taking sample of the patient blood and then they will try to get the amount of DNA of that kidney. That kidney belongs to somebody else, right? And everybody has got its own DNA composition. So if the amount of you know, other person's DNA, kidney's DNA is less, very less, it means that situation is under control. And if the amount of DNA is more and it's increasing day by day, it means what? The WBC has already waged the war and kidney cells are progressively dying and one day the kidney will fail. You get my point? So it is also being used to monitor that how this organ transplant is getting success or not. You get my point? So this three area you need to connect. CFDNA, cell-free DNA. Whole concept is what? That in my body, in your body, cells would be dying, fragmentization of the cell will happen, and the DNA will get free out of it. It will come into the bloodstream. Get it from the bloodstream and get the information about the DNA. Tumor in our own body. And tumor in our own body means what? It has got some DNA differences and that can be found out. So you can write cell-free DNA. These are fragments of DNA. These are fragments of DNA. These are fragments of DNA present outside the cell. Present outside the cell in our body fluids, such as blood, plasma, etc. Blood, plasma, lymph, etc. Blood, plasma, lymph, etc. Right. Next you can write CFDNA can be used in following ways. CFDNA can be used in following ways. First, non-invasive prenatal test. Non-invasive prenatal test. Right. And there you can write embryos DNA fragments. Embryos DNA fragments can be recovered. From mother's body fluid. To find out possibility of genetic disease. Possibilities of genetic disease in embryo. Right. Second, you could write to detect the cancer. At primary stage. To detect the cancer at primary stage by detecting the DNA fragments of tumor cells. DNA fragments of tumor cells in body fluid. Third, you can write to monitor success of organ transplant organ transplant. Right. Next one more. For detection of infection disease or infectious disease by detecting DNA fragments of pathogen. An infection agent is known as pathogen. DNA fragments of pathogen or infection agent. So let's say if some attack is there on the body, bacteria, virus, so bacteria will also be dying. It's not like bacteria will always survive because there is ongoing war going on between WBC and bacteria. So bacteria will try to kill WBC, WBC will try to kill bacteria, and both will be killed, right? It's like fight where the both party will get some damage. So if we take the DNA, blood sample out, in the blood sample, if we could find the DNA fragments of bacteria, then we can detect it with the disease only, right? So that is one. Now, there is another topic like we talked about, genome, or let's say genomics, right? So now genomics has got two things. One is genomics, normal study genomics, human genome project, Earth biogenome project. But now genomics has gone to two levels. One is called personal genomics. Right. Personal genomics. And another one is called metagenomics. Metagenomics, they have asked question in prelims. So personal genomics means one particular individual. If I get my genomics done, then it is called personal genomics. Custom genomics is also same thing. So either you call it personal genomics or you call it custom genomics. Right. So very simple to understand. The whole purpose of personal genomics is, right, individual genomic study. So if I go, you go for genomic study, it's a personal genome. Nowadays it's possible. $1,000 all you need to get it done, right? And it is done to detect the possibility of any disease, possibility of cancer. It's a lot many things that would come up. So when I say personal, it's kind of individual centric. Metagenomics is what? Meta means community. It's a community centric. It's like, eDNA. It's related to eDNA. eDNA means what? Environmental DNA. And in environmental DNA, you won't find only one DNA. You will find different species DNA. If you go to the soil, in a jungle, and in the jungle you get the soil, then in the soil you are going to get a lot many DNA. Lot many animals are living around. They are living their DNA in some form or other. And then you get the entire structure of the community, right? And if you get that, it is called metagenomics. What we call it? Metagenomics. So meta means community level genomic study. So genomics, genomic study at community level. You know community, right? Ecosystem, community. So community is what? Many population living together in a particular area, right? So that is community. Community means biotic factor. It's just simple as this. So in any ecosystem, all the biotic factor put together, that is known as community. Community along with abiotic factors like environment, that would make what? Ecosystem. So genomic study of community. Community means different, all population living in that area, right? In that area. And we have example of metagenomics, like let's say, if I say soil metagenomics, you understand now? Soil metagenomics. What do you understand? That means you've taken a sample of soil and then you study all the DNA content present in it. And then you get to know that which kind of animals are living around that or whatever information you want to get. Then we can get aerial metagenomics. That was a question that has been asked. Right. Aerial metagenomics. Right. Aquatic metagenomics. Aquatic metagenomics. Right. So personal genomics is, it's a kind of personal testing that you can go and do it. And this is for a study purpose. It's a again linkage with the environmental thing, right? So that is all about the DNA and genomic. That's one area. I mean, a lot of fundamental things are there. In the regular classes, we have gone in a length to discuss it, right? Now, we'll go to the next thing, and that is GM crops. Again, only those things which are important. One thing that you should be very clear as a UPSC aspirant, that GM crop is important for India, right? And we are going to accept it, right? If they ask one thing, that whether GM crop should be accepted or not, your take should be, it should be accepted, right? With all arrangement that its benefit should reach to every part of the society, right? Only one thing, there is a problem. Problem kya hoti hai? When there is a development must benefit all section of the society. That is what we make sure of, right? If there is development and all section of society is not able to enjoy its benefit, no matter which kind of barrier you create, sometimes price barrier is there. So that will lead to dissatisfaction, right? That will lead to imbalance, and deprived section of the society of that particular technology. Now, that section will become rebellion. That section become like, non-satisfied group, right? तो अभी वही हम कर रहे हैं. So GM crops में today, if we talk about today, so today there are two things which are going on, which are important. One is golden rice, and the second one is DMH 11, that is GM mustard. GM mustard. Why these two are important? Because Philippines became the first country to accept golden rice in this entire world. Bangladesh is at the last stage of the field trial. And in the coming time, within a month or a year, Bangladesh is going to be the second one to accept the golden rice. You get my point? So golden rice, you know that it's rice with vitamin A, right? And the gene responsible for it is called beta carotene gene. So what they have done, that they have introduced a beta carotene gene in the rice, right? And the rice is now will be there with the vitamin A, right? So beta carotene. The basic science behind this vitamin A is that we don't eat vitamin A. What we eat is beta carotene or carotene. Carotene is like, yellow pigments or orange red pigments. Like we say that mango is rich in vitamin A, papaya is rich in vitamin A, carrot is rich in vitamin A. We can say it like, but in a most precise term, we can say that papaya, mango, carrot, spinach, these are not rich in vitamin A. They are rich in carotene. And once you consume carotene, our liver converts carotene into vitamin A, right? So liver converts carotene into vitamin A. So carotene क्या है? Carotene is provitamin A. It's provitamin A or precursor of vitamin A. And liver converts it into vitamin A. That is why yellow fruits and vegetables are considered to be good source of vitamin A. I'm not suggesting that vitamin A is being created by the body. No, it's not. It's just like we consume beta carotene or carotene, and this carotene is just one step behind the vitamin A. One chemical change and carotene converts into vitamin A. And that chemical change would be done by whom? It will be done by liver, right? So carotene, provitamin A converting into vitamin A. So what they have done, scientists, they have done that in the rice genome, they have introduced something called beta carotene gene. So अगर हम equation के form में लिखें, like this, if I write it, that's, you will get it in a proper way. So I can write it like this. That rice genome plus beta carotene gene, and then become what? Golden rice. Because beta carotene is what? It's yellow in color, so the rice has got golden rice. And this rice will have starch plus vitamin A, both of the things. And that is just to counter the vitamin A deficiency. This has been developed to counter vitamin A deficiency. So the whole concept is important, that rice genome, beta carotene gene, golden rice. And because beta carotene is a little yellowish in color, so rice is now won't look white. It will look like golden after polishing and all, right? Second point you can write, Philippines became first country in the world. Philippines became first country in the world to accept, to commercially accept golden rice. Golden rice. In Bangladesh, last stage of field trial. Last stage of field trial is going on. Last stage of field trial is going on. Right? So that is why it is important. This is just to counter vitamin A deficiency in this entire world. Vitamin A deficiency may lead to death to the embryo, or let's say visual impairment, because rod cells in our retina that can only be constructed, we have enough vitamin A in our body, right? Sorry? Anticipation of changing the human DNA. That will change human DNA structure. I mean, it's anticipation only, right? But now you know that recently there was, I will talk about this, I mean DMH11, right? Let's talk about DMH11. You will get that answer too. So DMH11 is developed in India, right? It has been developed by, it's a GM mustard. It is HT variety, HT mustard. HT mustard means herbicide tolerant. Herbicide tolerant. You know herbicide? It's a kind of chemical, and these chemicals can be spread over the crop field, and this chemical will selectively kill only weeds and herbs, sparing the crop. है ना? So the crop productivity will increase because you are able to manage the herbs and weeds, the unwanted plants which is, there into the crop field. But then this herbicide chemicals cannot be used everywhere. Mustard में आप use नहीं कर सकते because mustard will also die. If there is a mustard plantation going on, and in mustard plantation along with the mustard there are some weeds coming out, then you know that weeds are naturally selected and they will rob all the nutrient of the soil. They will rob all the irrigation that you are giving, and they will grow at the faster rate. Sometimes weeds become longer than the crop. So it will create competition to the sunlight also, right? Some weeds are so dangerous that they secrete a kind of chemical around their root and then they kill the nearby plant. That is your crop. That is why in a healthy agriculture practice, we always try to control weeds growth. And the best way to control is to kill it chemically, because if you have long and very big farmland, then manually you won't be able to control it. If you have a chemical spray, you can spray it simply. But you cannot spray this chemical in a mustard plantation, because if you spray this chemical in mustard plantation, then weed will die and the mustard crop will also get damaged. तो फिर फायदा क्या है? So to deal with this situation, many countries in this world have developed HT mustard, herbicide tolerant mustard. I mean the mustard which is tolerant to that chemical. I mean, I just mean to suggest that in future, if you grow DMH11 in our croplands, and then you can easily manage the weeds because you spray the weedicide or herbicide, weed and herb will be dead and mustard will not be having any problem. Just by managing the weed, the mustard productivity can be increased by 30%, according to the government or let's say laboratory reports, right? Long back, GM mustard was there in India by one Germany company. So there was a German company who wanted to push GM mustard in India, in Indian agriculture system, right? But we denied because we have already accepted one, and that is called BT cotton. Today in India, there is only one crop that we are growing commercially, that is BT cotton, right? And BT cotton is a non-food crop. We are growing it. From 2002, we have been growing it. But we rejected GM mustard of Germany because of just one reason, that why should we accept all the things from the foreign origin? You got my point? I mean, BT cotton from America, GM mustard from Germany, and then the third would come from some other country. So we will be depending upon them for our seed supply. And then we wanted to develop our own, right? And we developed it. So it was developed in Delhi University, developed in DU. Dr. Deepak Penton. Although this question has been asked long back. UPSC के ऑप्शन में गलत था, पंजाब यूनिवर्सिटी. You just need to struck off, right? अब ये क्वेश्चन नहीं है कि DMH11 क्या है नहीं है, वो पुरानी बात हो चुकी, right? It's just like if you accept DMH11, then we are going to increase our mustard productivity by 30%. And we know that India is deficient in edible oil. We import a lot from outside. Then only we are able to meet our demand, right? Now, there is something called GESE you must be knowing. Genetic Engineering Appraisal Committee, and that is under Ministry of Environment, Forest and Climate Change. And they will take every decision regarding any GM crop. So according to GESE and Ministry of Environment, they have done environmental release of DMH11, right? So it's not yet released. It has been released at the environmental basis. That's a very clever thing the government has played. इससे पहले गवर्नमेंट क्या करती थी? इससे पहले गवर्नमेंट करती थी लैबोरेटरी ट्रायल्स, फील्ड ट्रायल and release, right? जैसे कि ET cotton, that is the only crop we have. So there has been lab trials, field trials, and after getting all the data, we just release it. But then the government has been trying to release it commercially from very long time. GSE has approved in 2017 में approve कर दिया था. GSE can give approval, it's a committee, but ministry will take the final decision. So ministry has taken decision in 2021. Let's release it at the environmental level. Now this environmental release will have two factors. It will serve two purposes. The first purpose, first, environmental impact. Study. Because there has been a lot of issues. They have been saying, like in a field trial where the DMH11 was being grown, honeybee population has gone down. Now it has been refuted by the study कि ऐसा कुछ भी नहीं हुआ. Then like the soil fertility will deplete. Then it demands more irrigation. That is true. Recently something came like grain size is very small, but doesn't matter because output will be more, right? So we have been refuting all the misconceptions or anything which came by that. And the second purpose of it is seed development. है ना? What we are going to do? Seed development. What is seed development? Let's say once you release environmental at the environmental level, the next step was commercial release. And to commercially release it, you need to create seed. So environmental release by the government is serving two purposes. One is environmental impact study to eliminate all the fear out of it. And the second one is that let's develop seeds so that in a next stage, when we go for a commercial release, we can supply the seed all through the market. तो ये चल रहा था और तभी क्या हुआ, इस पे केस कर दिया गया PIL, and the case is in Supreme Court. And the case is all about that it should not be released, right? Because it's a food crop, it will impact the environment, it will impact the human health, all kind of apprehensions are there, right? But the government has to say something different. पहली बात तो देखो GAC ने और MOEFFC ने एक्सेप्ट तो किया है ना. तो एक बात तो आपको यूपीएससी एस्पिरेंट की तरह समझना है. I mean, there is a lot of difference between UPSC aspirant and an activist, right? You have to choose just one, right? You may be activist in your real life, but when you are going to write the examination, activist को उस दिन घर पे बिठा दो. You stay there. I'm going to write the examination. Once I come back, I'll accept you again. है ना? Yes, you can do activism once you are in service. वहां करो. But यहाँ पे क्या ना? We are trying to get into the government. तो government decisions के साथ ही चलना है. Right? So, government of India has already made a statement because Supreme Court में मामला चल रहा है, and there is a lot of evidences and a kind of logics that is being given to the bench of judge. So, government has made an appeal to Supreme Court that please take decision properly. Because if you ban it, it won't be in national interest. अब सोचो इस तरह का अगर कोई स्टेटमेंट आ रहा है तो you can imagine. Why? Because we are heavily dependent upon imported edible oil. And at this particular time, we always talk about food security. So already we have given. It's a Food Security Act now. And next step of the food security, beyond food security, what is that? Nutritional security. So India now should aspire for nutrition security to the citizen. And you cannot provide nutrition security if you are dependent heavily on imported edible oil, import pulse and other food materials. अभी तो सोचो ना, पल्स में भी इंपोर्ट करते हैं, edible oil में भी इंपोर्ट करते हैं, तो कहां से होगा? Fats, I mean, when we say nutritional security, it means balanced diet. And balanced diet doesn't mean only carbohydrate. I mean only starch. We are more than sufficient in grains. but we are not sufficient in edible oils, we are not sufficient in pulses. तो गुड फैट्स भी तो है ना न्यूट्रिशन का पार्ट. प्रोटीन इज अ वेरी इंपॉर्टेंट पार्ट ऑफ न्यूट्रिशन. चलो प्रोटीन तो बाद में देखेंगे, पहले एक को तो देखेंगे. राइट नाउ द क्वेश्चन इज दैट विल इट हैम्पर आवर हेल्थ? इज इट गोइंग टू डिस्टर्ब आवर ओन जेनेटिक्स? तो ऐसा कुछ नहीं है, हमारे जेनेटिक्स को वो कुछ नहीं कर रहा. राइट उससे ज्यादा पेस्टिसाइड डिस्टर्ब कर रहे हैं. यू गेट माय पॉइंट? So if you have genetically modified crop and you are consuming it, it's not going to disturb your body mechanism. And if you have non-modified crops and it's pesticide laden, then it is disturbing more. That is why the cancer is there. Moreover, अभी इस साल के पहले, इसी साल गवर्नमेंट ने स्टेटमेंट दिया, इस साल के पहले तक मैं क्लास में बोलता था कि आप अगर फॉर्चून का या कोई भी एडिबल ऑयल कंज्यूम कर रहे हो इंडिया में, मस्टर्ड ऑयल को छोड़ के, बिकॉज हम मस्टर्ड ऑयल ठीक ठाक प्रोड्यूस कर लेते हैं, तो चांसेस आर वेरी हाई दैट यू आर ऑलरेडी कंज्यूमिंग जीएम ऑयल, बिकॉज द कंट्री फ्रॉम वेयर वी आर इंपोर्टिंग इट दे आर ग्रोइंग जीएम, है ना. एंड गवर्नमेंट वाज नॉट सेइंग एनीथिंग अबाउट इट. अभी जाके सुप्रीम कोर्ट में गवर्नमेंट ने अपना पत्ता खोला और कहा कि व्हाट इज इट, आई मीन what does it mean that you are going to ban it? If you are going to ban it, then it doesn't make any sense, because already that oil being imported to India, that is coming from the country who are growing GM crops. So in the from last 20 years we have been consuming the edible oil, and the edible oil is GM. तो अभी अपना ही क्यों बैन करना है ना. I knew that one day government will play this trick कि 20 साल से तो खा ही रहे हो ना, कुछ हुआ तो नहीं, तो अब इसको भी एक्सेप्ट करो. यू गेट माय पॉइंट? बट वही है ना कि देखो इसमें क्या है, एब्सोल्यूट कुछ भी नहीं है. यू गेट माय पॉइंट? नोबडी कैन टेल दैट इट विल बी 100% सेफ फॉर द ह्यूमन बीइंग. राइट इट्स जस्ट अ काइंड ऑफ रिस्क दैट वी हैव टू टेक. तो रिस्क आपको किधर लेना है? देखो गवर्नमेंट डिसीजंस ऐसे नहीं चलती. इट्स नॉट लाइक यूटोपिया कैन नेवर पॉसिबल. इट कैन नेवर बी पॉसिबल. One, you should compromise on a food security and nutritional security. And then you see that people are dying out of hunger. And a lot of money going out to procure the food. Right? That is one way. Moreover, you have to take 2 or 3 things in consideration. First, that your population is bound to grow. Right? And second, climate change will impact your agriculture output. तो क्या तरीका है? मरना तो ऐसे भी है कि नहीं? अगर हमने इसको एक्सेप्ट, इफ यू डोंट एक्सेप्ट न्यू टेक्नोलॉजी, देन व्हाट विल हैपन? हाउ कैन वी इंक्रीज द आउटपुट? आर वी willing to depend more onto the other countries in terms of the food requirements, right? Or to increase the food output of our own country, are we going to use a lot of chemical? It will again lead to problem. वो तो proven है कि problem है उधर. The second one is accept GM crop. BT cotton we have accepted just because the BT cotton has, given us a way to minimize the use of chemical pesticide in the crop field. So if less use of chemical pesticide is there, it means it is helping in environmental protection, it is helping in biodiversity protection, it is helping in our own health protection, right? This one is not for pesticides and all, but obviously it is for production. So GM crop is the necessity of the present time. The second green revolution, we are standing just before it. Once we get the GM crop open-heartedly, then the India will go through the second green revolution. First green revolution से क्या हुआ? फूड सिक्योरिटी दिया आपने इतने सालों बाद. सेकेंड ग्रीन रिवोल्यूशन से आप क्या देंगे? न्यूट्रिशन सिक्योरिटी, राइट? सो वी हैव टू चूज. वी हैव रीच्ड टू द स्टेज. आप एनवायरमेंट भी पढ़ चुके हो, सब कुछ पढ़ चुके. आपको लगा नहीं कि हम उस स्टेज में पहुंच चुके हैं जहां पे हमें दो बुरे में से एक बुरा को चुनना है. इट्स नॉट बिटवीन अच्छा एंड बुरा, राइट? देयर आर टू थिंग विच आर बैड. वी हैव टू एक्सेप्ट वन विच इज लेस बैड and we try to make it good. We'll constantly try to make it good. So that is why there is a problem with the GM crops. Obviously no technology will come without any problem, but this problem can be solved. We can take care of it, है ना? Yes, please. That is the one of the impact of the green revolution. Food तो है, but the thing is that that is true that we are growing surplus of cereals, but when it comes to pulses and oilseeds, then we are deficient. तो अभी क्या है कि हम, आप कभी सोचा कि ऐसा क्यों है? Why we are surplus in wheat and rice? एमएसपी मिल जाता है, फार्मर को पता है पैसा मिल जाएगा, राइट? बट पल्सेस एंड ओइलसीड्स, एमएसपी वहां पे भी है, लेकिन जेस्टेशन पीरियड ज्यादा है, देन क्रॉप सक्सेस होगा नहीं होगा, है ना? प्रिपरेशन ऑफ द क्रॉप लैंड्स, मैनेजमेंट ऑफ द वीड. आप सोचो ना, मैनेजमेंट ऑफ द वीड इज अ प्रॉब्लम इन मस्टर्ड. नाउ दिस प्रॉब्लम माइट हिंडर many farmers to accept mustard into the crop field. फिर वो क्या सोचेगा? वो तो बिजनेस की तरह देखेगा ना कि at the end of the day how much money I can earn out of it, right? हाँ, तो वो करना पड़ेगा. We have to create a conducive environment, and then we have enough thing. We can one day we can be self sufficient. बिल्कुल बिल्कुल. देखो, वही तो मैं कह रहा हूं ना कि न्यूट्रिशन सिक्योरिटी इज नॉट ए स्मॉल थिंग, इट्स अ वेरी बिग थिंग. आई मीन यू जस्ट सी वन थिंग, ग्रीन रिवोल्यूशन हुआ एंड देन फूड सिक्योरिटी बिल पास करने में कितने साल लग गए? रिसेंटली ओनली 1 decade बैक वी हैव डन इट. तो आपको क्या लगता है न्यूट्रिशन सिक्योरिटी अभी 2-4 साल में होने वाली है? कुछ नहीं, ऐसा नहीं. आप ही लोग करोगे जाके आगे. वंस यू बिकम सीन, 20 साल लगेंगे अभी, right? And in next 20 years we are going to explore all possible ways, fortified food, GM food, all the way we have to do it, and then only we will be able to do that. ऐसा नहीं कि GM potentially alone can do it. No, GM तो चलो, GM is a new technology that is being accepted, and the thing is that now at this particular point of time, if you don't accept GM, you will fall behind, because many countries have accepted it. Argentina, Brazil, America, Canada. Bangladesh is growing BT, brinjal, GM. I mean, already Bangladesh is already, I mean, they have given a kind of, decision that they are going to accept golden rice. Philippines has accepted it. Philippines to socho ki it's one of the key player in a rice research along with India, Philippines, China and Japan. वो भी accept कर लिया. Right. So this is what is important, DMH11, right? And the ongoing case is there, and that is why this entire thing has been important. One more thing, I mean, not go into the details that how DMH11 has been created, right? And for that, I can just tell you one thing, that DMH11 has been created by adding some gene from bacteria. So it was a kind of mustard plus 3 genes from soil bacteria, and that is what is DMH11, Dhara mustard hybrid 11. 3 genes from soil bacteria. Right. So this is about DMH11. One question has been already asked long back. Another topic that we should always talk about is GM rubber. Why GM rubber? Because recently India has become the first country in the world to plant GM rubber, right? So first GM rubber has been planted in Guwahati, Assam. Guwahati, Assam. Right. That's the world's first, है ना? World's first GM rubber. This was developed by Rubber Research Institute of India, Kerala. Rubber Research Institute of Kerala. Right. And why they have developed? To increase the productivity of northeastern state. That is why it has been planted in Guwahati. Right. Why? Because in northeastern state, winter is not suitable for rubber plantation. Rubber plant originally came from what? Amazon. And rubber is suitable for hot and humid condition. For rubber, it's hot and humid. That is why you see that southern part of India, they're good in rubber production. Sri Lanka is very good in rubber production. Any country which is around equator, where hot and humid conditions are there, they are good in rubber production. But northeastern states are also good in rubber production. लेकिन वहां rubber production 8 months ही होता है. So 4 months of winter there is no production. So what they have done, they have made it winter tolerant. Now this GM rubber will be able to grow in the same way that it is growing in a summer season. Right? So you can write one more point. Rubber is suitable for hot and humid condition. Suitable for hot and humid condition. But GM rubber can grow in a winter condition. The GM rubber can grow in winter condition as well. Winter condition as well. Right. So what they have done in one line, I can tell you that rubber, normal rubber, and with rubber they have added a gene called MNSOD gene. Sometimes they can ask the gene name also. And then we got what? GM rubber. So rubber genome plus MNSOD gene, and then we got the GM rubber. So conceptual clarity should be there. And this is only to increase the production. GM crop has different purpose, right? As a whole, if you look into, then GM crop has 4 main purpose. One is to increase productivity. The second one is to minimize the use of chemical pesticide to save the environment. That is why we have accepted what? BT cotton. Third one is to create herbicide tolerant plants so that we can manage the weeds in a better way to increase productivity, right? And the fourth one that we are doing to, create a kind of new, fourth one is to add some better nutrients into it. And the best example is what? Golden rice, है ना? So if we talk about 4 benefits as a whole of GM, then to increase productivity, and example is GM rubber. Second is herbicide tolerance, an example is DMH11. With everything we have one example. Then one is pest resistant. Pest resistant, that is called BT, cotton. And the last one is increase nutrition, and that is golden rice. That is why these all become important. Right. Although two more benefits are there, but ठीक है, उसको छोड़ के भी बढ़ सकते हैं. That is called new plant product and medicinal properties in the plant. But that is a far dream. I mean, we have to achieve this first. And we are focusing on this. आप देख रहे हो, GM rubber, we are already accepted. I mean, not accepted. The first plant has been, just single plant, एक ही plant है, 2021 में लगाया. DMH11, we are about to accept it. BT cotton accepted, and golden is lot the buzz are going. You know that there is one more thing, because you know entire Europe is closed for transgenic crop, right? They don't accept it. In 2021 only, France has claimed that India has exported to France transgenic rice, right? And France has a serious issue with that, because Europe is not open for growing, developing and consuming transgenic crop. Companies बनाती हैं, but वो खुद नहीं grow करती, right? India is one of the exporter to the European country where it comes to rice. हम rice को export करते हैं उनको, है ना? उससे पैसा भी मिलता है. But now our own agriculture system is under threat. Threat कैसे हो गया? So you know that there is something called field trial, So field trials. In field trials, what we do, that our own government and research organization like, agriculture research institutes and all, they get in association with farmers. And with farmers, we do what? Field trials. We provide seed to the farmers, technical assistance to the farmers. Farmers will grow it like the way they grow their own crop. And once the crop matures, we buy back all the thing. We don't allow this trials thing to go into the market, because it's a trial. We have to test it. But slowly what happened, because in India, these trials are going on in last 2-3 decades. 3 decades हम try कर रहे हैं. And more than 20 crops are under field trials, right? So now farmers in last 2 decades, they could see visibly that if they are growing this GM crop, then crop productivity, crop quality, crop is better. And when they are growing the normal one, it's not that good. So what happened in India, that at many places, seed of the field trial has been taken illegally, and now it is being practiced into the normal cropping. You get my point? Like entire Punjab, Haryana, Andhra Pradesh, they are developing BT brinjal. I mean, if you are in Delhi and you are eating brinjal, it may be possible that you are eating BT brinjal, which has not been accepted by the government, right? And similar thing happened with the rice also, because we are having some rice, GM variety of the rice is also under field trial, and we buy back, we destroy it, right? But farmers has now illegally taken some of the seed of the rice, and now this GM rice is also being grown in India. And that is how this entire rice that we are exporting, certain percentage of the rice is basically GM rice. अब ये illegal farming है. For that, under Environmental Protection Act, there is provision to, punishment and all. But क्या कर सकते हैं? Because government is also not taking very strict action on that. तरीका भी नहीं है. Farmers are unaware. They have done it, and it's so widespread that it's very difficult to control. In Haryana, there was a BT brinjal crop going on, and the government wanted to destroy the crop. There was a kind of protest between police forces and the farmers, and they could not do anything. तो हमारे लिए लेकिन प्रॉब्लम क्या है ना, कि our agriculture systems are under question. So we came up with something called non-transgenic GM crop. Non-transgenic GM crops. GM crop technique. Or it is also called SDN technology. So again, it's a recent one because it has been recently approved by GAC. Non-transgenic GM crop. You know that GM can be of two types. So if I say GM, genetically modified, it can be two types. Transgenic. And then it could be non-transgenic. Just understand it. When I say transgenic, it means what? In transgenic, it will be like transfer of gene between unrelated species, right? So I will not go in definition and all. Let's symbolically understand. So species 1 and species 2, and then we get what? Transgenic gene. That means gene from a species 1 is transferred to a species 2. Two different species. You got my point? Let's say beta carotene gene has been transferred to rice. So beta carotene gene kahan se nikala? Daffodil se. Daffodil is another species transported to rice. So it's transgenic. You get my point? It's simple like that. If you transfer a gene from mango to lychee, it is transgenic. Species has changed. If you change the species, technically, if you look into that thing, risk is more. Right? Longer field trial are needed. Longer risk assessment are needed. Because you are transferring the gene from bacteria to plant. In BT cotton, what we have done? We have transferred a gene from Bacillus thuringiensis bacteria to cotton. So bacteria and plant, it's transgenic. Anything could be possible. You can transfer a gene from plant to bacteria, bacteria to plant, animal to plant, plant to animal. कुछ भी कर सकते हो. UPSC asks one question, basic one. Which of the following can be possible in recombinant DNA technology? Everything is possible because DNA structure is same. DNA structure is same, right? No matter which building you talk about in this entire Delhi, the internal of the wall is same. It's made up of bricks only, right? So no matter from where you take the brick, you can put it anywhere. यहाँ का brick कहीं लगा दो, वहाँ कहीं लगा दो. So if they ask such possibility कि where we can do change, where we cannot do change, nothing is, impossible. You can make change. You can transfer the gene of plant to human being. Technically, you can transfer. What kind of benefit we'll get, we don't know. But transfer is possible, right? So if you do such transfer, it is called transgenic. But non-transgenic means what? We don't transfer the gene. We remove the gene just, right? We have one rice and we removed few gene of the rice. After removal of the gene, the rice variety is improved. Because we have removed something which is not of our own use. Right? Sometime we can do it. Let's say, plants have their own genetic composition and grain size is just one part of the plant. है ना? हमने क्या किया? हम तो natural selection पे नहीं जाते, हम तो human selection कर रहे हैं, artificial selection. We are just looking for the mango should be there and mango should be good in taste, good in smell and good in size. Plant तो ऐसा नहीं सोच रहा ना. Plant का क्या है मतलब? Plant तो survive करना चाहता है. Plant is all about that I should produce seed and the seed should survive and the next generation should come up. You get my point? But we focus on the product. So we do one thing. We know that plant is utilizing its internal resources in different things. वो लीफ भी बना रहा है, स्टेम भी बना रहा है, ग्रो भी कर रहा है, फ्रूट्स भी बना रहा है. तो कुछ जीन्स को काटा देते हैं ताकि कुछ एरिया उसका कॉम्प्रोमाइज हो जाए, उधर रिसोर्स वेस्ट ना हो और रिसोर्सेस कहां चला जाए? फ्रूट्स में, जो हम खाएंगे. You get my point? So you can modify in a two way, by inserting a gene that is called transgenic and by removing some gene that is called non-transgenic. Europe has problem with what? Transgenic. Europe has no problem with non-transgenic. Right? You get my point? There is a difference between genetic modification and transgenic. GM can be of two types, non-transgenic and transgenic. Modification तो आपने किया ही ना. You can modify by adding a gene and you can modify removing a gene. So here what is there? Removal of gene from plant genome by an enzyme that is called site directed nuclease, that is called SDN, site directed nuclease, SDN. इसका एग्जाम्पल क्या है? BT cotton, that only thing that we have. But we don't have example of non-transgenic. We are yet to develop it, but it has been accepted, right? So now you understand what is non-transgenic one. SDN, site-directed nuclease. Site-directed nuclease. SDN. That is why it is called SDN technology, right? Next, you can write. In India, this technology has been developed by. In India, this technology has been developed by Indian Agriculture Research Institute. Indian Agriculture Research Institute. And it has been accepted by GEAC. Accepted by GEAC. GEAC, right? Next you can write European countries are not ready to accept transgenic GM crops. But they can accept non-transgenic GM crops. Non-transgenic GM crops. Right. Next you can write non-transgenic GM crop. Non-transgenic GM crop are comparatively safer, and it require less field trial and risk assessment. Less field trial and risk assessment. Right. So longer field trial will not be required. It will be less duration. Risk assessment will also be less because we are not adding any gene. We are just removing it. Scientists have already considered that non-transgenic is natural. It's very close to the natural process of mutation. In mutation also sometimes some gene eliminates, right? The whole purpose is that eliminate those genes of the plant which is not of our own use and then we can make it in the way. है ना? जैसे कि आपने देखा होगा कि nowadays the plants has gone, low in their height, है ना? तो पर्पसली हाइट कम किया सबका, so that उसकी जो टोटल टाइम पीरियड, मैच्योरिटी टाइम पीरियड कम हो जाए। नॉर्मली जो क्रॉप आज से 10 साल पहले 4 महीने में ग्रो करते थे, आज 3 महीने में करते, राइट। ऑल्दो इट्स नॉट नॉन ट्रांसजेनिक, क्रॉप के डेवलपमेंट में भी हमने बहुत सारे काम कर रखे हैं, है ना। यू नो दैट देयर इज समथिंग कॉल्ड न्यूक्लियर एग्रीकल्चर। So in that only we can talk about nuclear agriculture. Yes, please. अभी तक डेवलप नहीं किया. It's just recently in 2022 only GSE has approved it. So we have yet to get one non-transgenic one. No. So nuclear agriculture is development of new crop variety with the help of radiation, gamma rays, leading to mutation. Leading to mutation. So what we do, we use gamma radiation, and you know Baba Atomic Research Center, they are not only engaged with the nuclear energy, rather they have already developed more than 45 types of crop varieties. And many of the crop varieties have already made its way to the crop field, right? So gamma radiation are most dangerous one, you know that. It has got the highest penetration value. And on a seed, we throw gamma radiation, and then genetics of the seed will be changed. It's not like targeted one, it's like hit and trial method. You get my point? वहां भी वही मामला है, वही नॉन ट्रांसजेनिक टाइप ही है. हालांकि नॉन ट्रांसजेनिक इज एंजाइमेटिक मेथड, यू नो. देयर वी आर यूजिंग व्हाट? साइड डायरेक्टेड न्यूक्लियस. न्यूक्लियस इज समथिंग व्हिच कैन कट द जीन आउट. Whenever we say nuclease, it's something like gene cutter. Nuclease. When this term nuclease comes, it like molecular scissors. Right. It's cut the gene. But here, and whenever we talk about enzyme means always biotechnology. Let's say if there is a crop improvement and we are developing new crops. And if the crop has been developed with the help of radiation, and one crop has been developed with the help of enzyme where the gene has been cut or gene has been pasted. So जहां भी enzyme use होगा, it's a biotech. Wherever you use radiation is a nuclear technology, right? So here this is nuclear technology. This gamma radiation will hit onto some gene and it will destroy the gene. Once the gene is destroyed, it means some character of the plant has been eliminated. And the plant will show, it may show some character which are in favor to us. You get my point? But one thing is there that biotech are targeted one. You know that we have to cut this gene and this gene will give this kind of impact. But in nuclear technology, it's not so targeted. It's like hit and try. You do on 500 seeds, we grow it all, and then you select one of them. But in that method only, like Baba Atomic Research Center, you can write Baba Atomic Research Center has developed more than 40 types of crops by this method. More than 40 type crops by this method. Many oilseed and pulses has been developed. Many varieties of oilseeds and pulses has been developed by this method. right? So that is about agriculture part. Now we'll move on to the next one. And the next one is health area, right? So biotechnology in health sector. And in health sector, the main issue is moving around your, you can say like gene therapy, right? So there is something like a stem cell, but a stem cell is again, there is nothing new has come up into the stem cell. You just need to brush up any guide, any book, any notes that you have, you go through the stem cell. It's an important one because they can ask question from a stem cell basic concepts. Stem cell has got a lot of basic concept. You should not, I mean, this class doesn't mean that you just need to cover this class only. It means that you have to cover all the things that you have done so far, right, in the preparatory journey. It's just a kind of that we are drawing your attention towards some of the issues related to current related aspect or the important one, right? Because during this time, I always get many query and questions on social media, WhatsApp, Instagram, har jagah. And people will ask for, sir, I'm writing prelims examination. Please indicate some important topics of science and technology. I'm sure that no teacher would give you reply if you ask this question. Anna. So like two questions are there, good morning, and the second one is this. I simply say good morning, baat khatam. है ना? क्योंकि क्या ही हो सकता है? I mean, it's impossible to guess, right? It's impossible to guess. No teacher can tell you in any subject कि ये वाला पढ़ के जाओ, ये important है. I mean, it means that the moment you say ये वाला पढ़ के जाओ important है, दूसरा कुछ पढ़ेगा ही नहीं. And it's not like that. UPSC has a random way to ask the thing. So there is nothing like, I mean, there has been time when question was totally in a different tangent. And so nothing work in that way. Anyway, so there is something called, let's say, gene therapy. That's health related thing. And gene therapy means, let's say, if we could find that in our body there is one gene and this gene is responsible for cancer, this gene is responsible for a particular disease, then scientists are now trying to develop some way by which they can eliminate this gene out of the genome to remove the possibility of the disease in our body or in the next generation, right? So there could be two ways. Let's say if I am suffering from a disease due to some genetic aberration, then the gene in my body can be replaced or removed so that I become healthy and rest of my life will be healthy. But again, one more thing. Let's say if my beta cell, my pancreas is not producing insulin because there is some problem with the pancreatic cell. So they have placed some gene inside the pancreatic cell and now this new gene, which are healthy one, will produce insulin and my insulin will be reverted back to normal. So I am now free of diabetes. Technically it is possible. Practically it has not been done. But then diabetes has been eliminated in my body because you acted upon the pancreas, which is source of insulin, right? But in my next generation, I'll be contributing the genes through a sperm or a lady will be contributing a gene through ovum. तो वहां तो वो खराब जीन जाएगा ही क्योंकि चेंज तो आपने किया किस में, बॉडी के पार्ट में. बट a sperm is being produced by the testis or ovum is being produced by ovary. It means that this approach will give benefit to me only. Next generation will still suffer from that disease. है ना? तो एक दूसरा तरीका भी है and that is called germ cell gene therapy, where you tweak the thing, where you change the thing at the sperm level, ovum level or zygote level, right? But then it is controversial. You cannot do it. In some cases, it is allowed to experiment on the human body, but it is not allowed to experiment on the germ cell. You get my point? Why? Because success rate of gene therapy is not very good. And when I say success rate is not very good, it means the chances of error is very high. if somebody is terminally ill, if some person, adult person is terminally ill by cancer, we can take chance on that person, right? We can do a lot of gene therapy experiments on that because we know that the person is anyway going to die. So let's take a chance. If it pays off, it's all fine. It doesn't, then obviously we'll learn out of it something. But you don't want to take chance with the generations to come. Because if you make any change in a sperm, ovum or zygote or embryo, then it's not only going to impact that embryo. Rather, it is going to impact all the generations to come. You get my point? If anything goes wrong. That is why worldwide, germ cell gene therapy is what? Banned. तो क्लियर दिमाग में रखना है कि germ cell gene therapy is banned. No country can allow germ cell gene therapy. हमें करना ही नहीं. Just because it's not something which is going in a proper direction. Now the second one is called somatic cell gene therapy. Somatic cell means body cell gene therapy. That can be done, but in India, all the somatic cell gene therapy is being done as experiment, not as treatment. You get my point? So there is a treatment and there is experiment. Treatment means हॉस्पिटल में जा रहे हैं तो जीन थेरेपी डिपार्टमेंट लिखा हुआ है. किसी हॉस्पिटल में देखा आज तक जीन थेरेपी डिपार्टमेंट? नहीं है. It's like in a research center, there are gene therapy research centers. There is no department. No matter which hospital you go into the India, you will never find gene therapy department. Right? Because it's not treatment methodology, accepted treatment methodology in India. If anybody is willing to go through the gene therapy, it's experiment. An experiment will be done only when the doctor has approval from ICMR, Indian Council of Medical Research, in authorized research centers like AIIMS and other big establishment, right? But then gene therapy has a very bright future. In a coming time, we are going to use it, right? So gene therapy approach mein, there are two ways. One is called knockdown approach or knockdown method and knockout method. So the whole concept is that in a person's body, there is one gene and that gene is faulty. The gene is bad. The gene is responsible for a disease. You got my point? Now we can do two things with the gene. Knockdown का मतलब होता है gene silencing. Gene silencing. Right. And here what we do? Gene is not removed, but its expression means protein synthesis, is stopped. How? We'll talk about this. Don't worry. That is called knockdown. So gene is not removed, gene is there, but what we have done, we have blocked its expression, protein synthesis. अभी हमने शुरू में बात की थी ना, that gene has only one thing to do, that is to make protein. If there is a gene, it is going to make protein. If the protein is faulty, the body will have disease. We are diseased because of our protein structures. We are healthy because of our protein structure. If we get good protein, it's a healthy. If we got bad protein, it's unhealthy. Even the cancer is being produced into the body, then there are some change in gene and the protein it is being produced. We'll talk about that too. So that is called knockdown approach, gene silencing approach. Example, we can write, that is called RNA interference technology. RNA interference technology, right? Knockout approach का मतलब होता है जीन रिमूवल. right? So gene is removed from the genome. So gene is altogether removed, eliminated from the genome. And the best example of this is CRISPR-Cas9. I'm sure that CRISPR-Cas9 you might have heard about it. It's very much, I mean, it's a very important topic, CRISPR-Cas9. So knockout means just remove the gene, eliminate the gene. Knockdown means don't eliminate the gene. Gene will be there, but expression will be blocked. Like I always give the example in the classroom that in Indian system, sometimes criminal is hanged. So that is knockout. And sometimes criminal is put behind the bar. That is knockdown. That means criminal will not be able to express itself. So that's a knockdown approach, right? And sometimes you just kill the criminal so that there is no slightest of possibilities eliminated, right? दोनों बढ़िया है, लेकिन बढ़िया में यही लगता है, right? But gene silencing is also gene silencing. Now you got what is the meaning of gene silencing, right? If I talk about, तो अभी कहा था ना DNA, and DNA is basically gene, and DNA will make what? RNA, and RNA will make protein. Now let's think in that way. This gene is faulty gene, है ना? अभी क्या है, लोग इसको मानते हैं कि this is bad gene, faulty gene, gene for disease. Now this is gene for disease, right? And this is protein. It's not a good protein. It's a bad protein. And this bad protein is responsible for what? Disease. That is due to this protein, the disease is there. Now you can see, like at the expression level, it's protein which is creating problem. But at the root level, which is creating problem? Gene. है ना? So if we remove this gene, अगर हम इस जीन को हिट करें, then it is called knock out. It's CRISPR-Cas9. You got my point? Gene ही खत्म. No gene, no protein, isn't it? But if we hit onto the RNA, if we hit onto this, then it is what? Knock down. And that is called RNA interference technology. In both of the condition, what will not be formed? Protein will not be formed. Now, what RNA does? You must be knowing that RNA is nothing, it's a messenger. RNA is what? Messenger. DNA is there inside the nucleus. DNA will never come out. DNA will never engage itself into protein synthesis. DNA is a kind of, somebody who will only give instruction, right? DNA is like CEO of a company, right? And CEO will never engage in any kind of production. Rather, CEO will give information and command to manager or messenger. And manager or messenger will take this message of CEO to the factory, to the workstations, and then the product will be made, isn't it? So this is CEO and this is manager or messenger, and there the protein will be formed. In our cell structure, it's like nucleus. Nucleus has got the DNA. Then DNA will give command to RNA. RNA will come out of the nucleus, go to the cytoplasm. In cytoplasm, there are ribosome, and ribosome will make protein. Who makes protein in our body, in our cell? Ribosome. You know that. Ribosome is a machine which makes protein in our body. We know that. But ribosome is not making protein randomly. Ribosome will always wait for RNA to come and RNA to bring information from the DNA. That is why RNA is called messenger RNA. RNA is the bridge between protein synthesis and DNA. You get my point? Now, if you hit onto the DNA, you eliminate the DNA or the gene for disease, it's knockout. Let's leave the gene there only and you hit onto the RNA. Every time the DNA is passing on the message, you destroy the message. So, ribosome will not get the message and protein will not be formed. Disease will not appear. Even if you have disease gene in your body, the expression of that disease gene will not be possible because you are interfering with RNA. है ना? यही दो है. तो इसमें क्या है? जीन किल कर दिया, इसमें जीन को साइलेंट कर दिया, एक्सप्रेशन को साइलेंट कर दिया. यू गेट माय पॉइंट? एनी प्रॉब्लम? नाउ यस प्लीज. नो, दिस ऑल डिपेंड्स अपॉन. एक तो पहली बात है कि साइलेंसिंग से कुछ भी नहीं हो सकता. साइलेंसिंग इज ऑलवेज प्रेजेंट जनरेशन. क्योंकि साइलेंसिंग में कर क्या रहे हो आप? यू आर नॉट इंटरफेयरिंग विद द जेनेटिक मटेरियल, यू आर इंटरफेयरिंग विद द मैसेज. तो जेनेटिक मटेरियल तो वही रह गया. यू डिडंट चेंज विद द डीएनए. तो जीन साइलेंसिंग इंपैक्ट विल नेवर एवर पास ऑन टू द नेक्स्ट जनरेशन. रादर जीन साइलेंसिंग इंपैक्ट might lose after certain period of time. ये भी नहीं कि हमारे बॉडी में भी हमेशा रहेगा, है कि नहीं? क्या भरोसा, वो जेल में बंद कभी तो निकलेगा, है ना? ऐसा थोड़े ना कि बोल रहे हो पलटवा दिया आपने, है ना? तो वही मामला है कि जीन साइलेंसिंग का मतलब हो गया कि इट माइट रिवर्ट बैक. इसलिए जीन साइलेंसिंग ह्यूमन बीइंग में हम बहुत इस्तेमाल नहीं कर रहे, प्लांट्स में करते हैं, राइट? बट ह्यूमन बीइंग में कुछ भी नहीं कर रहे हैं बिकॉज टू डेंजरस टू यूज इन ह्यूमन बीइंग, बिकॉज एंटायर कॉन्सेप्ट इज वेरी डेंजरस, राइट? ऑब्वियसली वी वांट टू ट्रीट सम डिजीज, लेकिन मामला एक इंसान, देखो एक सिंपल सी बात है, मेडिकल साइंस में इंडिविजुअल ह्यूमन बीइंग्स की कोई वैल्यू नहीं है, राइट? दे ऑलवेज टॉक अबाउट ह्यूमैनिटी, है ना, कि दिस डिजीज मस्ट बी ट्रीटेड फॉर ह्यूमैनिटी. In AIMS and all research centers, they always look for someone who has a unique disease. अगर unique disease वाला इंसान मिल गया, that's a resource for them. They will experiment on them to understand and to develop new medicine. Anyway, तो अगर RNA पे अगर आपने एक्ट किया, तो it is always localized, is always to the present generation, and it's not even guaranteed that पूरे लाइफ टाइम रहेगा कि नहीं रहेगा. Why? Because here we destroy this RNA with the help of another RNA. That is why it is called RNA interference. So we make an RNA in laboratory, we put inside the body, and this lab-made RNA will interfere with the normal RNA, and then it will keep on destroying. But कभी हमारा जो बाहर से भेजा RNA वो भी डिस्ट्रॉय हो जाएगा, तो फिर से वो बीमारी आ जाएगी. In DNA, if we destroy it, it's all gone, है ना? अब क्या है कि DNA में it may be possible that it may be one generation or it may be all future generation. तो यहां पे क्या कर सकते हैं? If you want to add more concept into this, so this knock down, knock out, it can be of two types. One is called somatic cell, another is called germ cell. Somatic cell only present generation, only present generation, and germ cell all future generation, all future generation. It's approach where you do it. तो where germ cell में कहां करोगे आप? Most suitable thing is zygote. When the father's sperm and mother ovum has combined, fused with each other, and first zygote is formed, at that time only you eliminate the gene. तो बच्चे में भी नहीं आएगा, और बच्चे में कहीं नहीं आएगा. अब वो बच्चा अगर मेल है तो स्पर्म में भी नहीं आएगा नेक्स्ट जनरेशन के लिए, और अगर फीमेल है तो ओवम में भी नहीं आएगा. बट देन इट्स टू डेंजरस. ये क्या होगा? इट इज ऑलवेज लिमिटेड टू प्रेजेंट जनरेशन. बिकॉज ये तो जर्म सेल में ही हो सकता है और कुछ नहीं हो सकता. No, it's only to take out gene. CRISPR-Cas9 is a kind of seizure system. It's a cut. And for that matter, if you want to add some gene, you have to cut again. Add करने के लिए भी cut ही करना है ना? Because the addition is easier, cutting is difficult. You get my point? In biotechnology, if you want to cut something, it's more difficult. If you want to add something, it's an enzymatic process. You can add it. है ना? So biggest obstacle in gene therapy was that we wanted to have a very precise molecular seizure. है ना? And now we have got the most precise molecular seizure to cut the gene precisely from the genome, and that is called CRISPR-Cas9. That is why CRISPR-Cas9 is now at the present time considered as one of the biggest breakthrough in biotechnology. है ना? आप सोचो ना, we are able to develop GM crops. GM crop में भी तो जीन काट ही रहे हैं, जीन काट हटा रहे हैं, जीन जोड़ रहे हैं, यही तो कर रहे हैं. But in GM crop we are doing 1000 attempts to get the one right one. 1000 अपने चांसेस लिए, एक उसमें से सक्सेसफुल हुआ, आपने उसको एक्सेप्ट कर लिया. बट यू डिस्ट्रॉयड 999 क्रॉप्स इन दैट वे. वहां तो एफर्ट कर सकते हैं. कैन वी डू द सेम थिंग इन ह्यूमन बीइंग? कि कैंसर के इलाज तो होगा, लेकिन चांसेस क्या है? 1000 में एक का हो सकता है. तो लेट्स किल 999 टू गेट वन पर्सन. तो वहां पे तो हमें क्या चाहिए? व्हेन इट्स कम्स टू आवर ओन लाइफ, एवरी लाइफ इज इंपॉर्टेंट, एंड फॉर दैट वी नीड व्हाट? मोस्ट प्रिसाइज cutting technology. So at the present time, the most precise cutting technology is what? CRISPR-Cas9. It can accurately cut the gene, selectively and accurately cut the gene that we want to cut without disturbing anything around. And if you precisely cut it, you can precisely add it also. अभी तक human genomics का जो human genome therapy का मामला है, it's not about adding gene. It is only about removing genes. Right? So here again, two approaches can come up. One is that we will remove a faulty gene to eliminate the possibility of a disease. That is the first one we are trying to achieve with the help of CRISPR-Cas9. And second one, that we can replace the faulty gene with the healthy one. ये तो बाद में आएगा. You get my point? That's a future technology that might come up. Right? But then I'll tell you one thing, that this entire thing, idhar wala to pura controversial, because that also create a kind of controversy of designer baby, right? Because human beings are very difficult kind of animal, very difficult to manage them. So the moment they saw the possibility that genes can be eliminated and new gene can be added, on paper to possibility hai. So human being forgot about the disease. They were like, चलो ठीक है, फिर आंखें नीली कर दो, है ना, बच्चे का लेंथ बढ़ा दो, बाल घुंघराले कर दो, because genetic changes can do such thing. So gene therapy will become what? Cosmetic gene therapy. Like the way we have converted plastic surgery into cosmetic surgery. And that is one reason that we have stopped it. हमने कहा उधर काम ही नहीं करना, लैब तक ही रखना है. Because the moment we realize, and one day it might be possible, you never know. Many things that has been very difficult thing, nowadays it's a very common one. And the moment we realize that this could be possibility in future and easily it can be done, then you will see that many people with a lot of money will go to the doctor to get the desired traits and characters in their baby. Problem आएगी. Anyway, हाँ जी. विच टेक्नोलॉजी? आरडीएनए टेक्नोलॉजी. हाँ, जीन एडेड. डीएनए टेक्नोलॉजी तो क्या? डीएनए टेक्नोलॉजी तो पूरा ही वो के एक होता है डीएनए रिकॉम्बिनेंट टेक्नोलॉजी. हाँ, रिकॉम्बिनेंट में करते हैं. इट्स अ डीएनए रिकॉम्बिनेंट टेक्नोलॉजी, तो रिकॉम्बिनेशन ऑफ जीन्स. बट डीएनए रिकॉम्बिनेंट टेक्नोलॉजी कहां कर रहे हैं? वी आर नॉट डूइंग इन ह्यूमन बीइंग अगेन. We are doing in bacteria. We are doing in croplands. Like insulin is being produced by DNA recombinant technology done in bacteria. तो वहां पे क्या किया हमने? We take the human gene out of the human genome. We inserted this gene into the bacteria. But in order to insert one gene from human genome to bacteria, we might have killed thousands of bacteria. लेकिन कोई इसका ख्याल रखता है बैक्टीरिया का? है ना? ठीक है, मर गए. वन बैक्टीरिया वी गॉट इन अ डिजायर्ड वे, उसको एम्पलीफाई कर लिया, उसको मल्टीप्लाई कर लिया, और आज पूरे वर्ल्ड की इंसुलिन सप्लाई कौन कर रहा है? वही बैक्टीरिया कर रहा है जो पहली बार बना था. यू गेट मी पॉइंट? सो दैट्स डीएनए रिकमेंड, बट वी कैन नॉट डू अदर वे. अगर हमें ये पता चल जाए कि बैक्टीरिया के इस जीन को ह्यूमन बीइंग में डाल देंगे कैंसर होगा ही नहीं, it's very difficult to do it because we never know that how many attempt would we needed to do it successfully, है ना? Although there has been controversy in few years back, a Chinese doctor whose name is Zhang Kui has gone against world order to genetically modify twin babies, twins को modify किया by adding some gene into them. And now this twin baby is resistant to HIV. और बच्चे का जन्म भी हुआ. But that is the only case that happened in China. And the doctor and China both were criticized all over the world. Why? Because you are going against world order. वो तो अच्छा है कि वो बच्चे का जन्म हो गया और बच्चा जो है वो हेल्दी है. अच्छा, हेल्दी है या नहीं, कौन बता रहा है? चाइना बता रहा है. तो कितना भरोसा कर सकते हैं भी सोच लो आप. राइट. Rather it was Chinese, मतलब लोग तो ये भी कहते हैं कि कोई चाइना में कोई डॉक्टर वर्ल्ड ऑर्डर के खिलाफ जाके कुछ कर सकता है क्या? कर सकता है? अनलेस गवर्नमेंट एफिलिएटेड. इंडिया में तो कोई कुछ भी बोल सकता है, आपको पता ही है. यू कैन ऑलवेज गो टू सुप्रीम कोर्ट, राइट? बट चाइना में यू कैन नॉट डू एनीथिंग अनलेस गवर्नमेंट सपोर्ट्स यू. कोई नहीं, जो किया होगा गवर्नमेंट के सपोर्ट में किया होगा. वुहान में ये पता नहीं क्या क्या कर रहे थे, है ना? वैसे ही कुछ कुछ कर रहे हैं. दे आर वर्किंग ऑन एवरी फ्रंट. देयर इज अ लॉट ऑफ अंडरग्राउंड एक्टिविटीज गोइंग ऑन इन चाइना एंड दैट इज वन ऑफ देम. सो चाइनीज गवर्नमेंट ने क्या किया? जनकोई को बैन लगाया, 6 महीने जेल में डाल दिया. वो सब कुछ किया हल्का फुल्का जो करना चाहिए था. एंड इट्स नॉट लाइक स्ट्रिंजेंट एक्शन. इट्स जस्ट लाइक ठीक है, यू डन इट गुड, you have done it well, no problem with that. At least now China has entered into the book of history that first baby born with the gene therapy or genetic modification was in China. They knew that one day somewhere in Delhi UPSC class में कोई टीचर पढ़ा रहा होगा, है ना? और हर जगह पढ़ाएगा आप. So that is how they do it. Anyway, we'll move on to the. अब इसमें ये बताओ कि I have given all the. अभी this is something which is very lengthy one and two technology, CRISPR-Cas9. मैं थोड़ा थोड़ा बता देता हूं, not the entire one. You can write CRISPR-Cas9. CRISPR-Cas9 you can write. It is most precise and accurate tool for genetic modification. For genetic modification. Right. Next you can write CRISPR-Cas9 is naturally present in bacteria as immune system against virus. Immune system against virus. Immune system against virus. Virus, yes. CRISPR-Cas9 has 3 components. तो एक बात बताओ, CRISPR-Cas9 किसके अंदर है? इट्स इन बैक्टीरिया. We discovered it in bacteria and now we are using it for human being. So CRISPR-Cas9 belongs to whom? Bacteria. And the first one is CRISPR, that is DNA segment of bacteria, right? Second one is Cas9, that is called endonuclease enzyme. And nuclease क्या क्या बोला था? Molecular scissor to cut the gene, to cut the DNA or gene. So whenever nuclease, it's cutting instrument. And the third component, it's not there in this name, is called gRNA, guide RNA, guide RNA. So it has got 3 components. Yes, please. Endonuclease, exonuclease, endonuclease can be there. If this is DNA and you want to cut this part, then you have to use enzyme called endonuclease. अगर टर्मिनल एंड से एक तरफ से काट हटाना है तो क्या चाहिए? एक्सोन्यूक्लेज, राइट? You just need to catch nucleage, right? So these 3 things are there: CRISPR, Cas9 and gRNA. I'll just explain a little bit. क्योंकि देखो CRISPR-Cas9 से क्वेश्चन तो पूछे ही है. Now UPSC do what? That once they start asking any question, then initial questions are very easy one, right? And then they start getting into deep of it, है ना? Like the way we explore a chapter, शुरू में हल्के हल्के, फिर धीरे धीरे उसमें डीपली चले जाते हैं. Similarly UPSC will go. Like now the thing is that being a teacher, I am looking question paper since last 17 years. हर साल आ रहा है, हर साल देख रहे हैं. Being a student, आप कितनी भी कोशिश कर लो, उस होराइजन में नहीं देख पाओगे. You get my point? Although we always do partial question paper, लेकिन फिर क्या होता है ना, you are burdened with a lot of things. साइंस भी, environment भी, हिस्ट्री भी, geography भी. So that is where the specialized teacher who is teaching just 1 or 2 subjects, they have to focus on that. So we are able to see that. Like the way we read something, जैसे अभी क्वांटम में पूछा क्यूबिट्स क्या है? क्यूबिट्स इज द मोस्ट बेसिक कॉन्सेप्ट ऑफ द क्वांटम कंप्यूटर. मतलब क्वांटम कंप्यूटर किसी ने आर्टिकल कभी पढ़ा होगा, क्यूबिट बिना पढ़े वो आगे बढ़ा ही नहीं होगा, राइट? बट नाउ क्यूबिट तो नहीं आएगा दोबारा, अब क्यूबिट के बाद वाले आएंगे. तो वी नो दैट क्यूबिट के बाद में, या आप भी जानते हो जब आपने पढ़ा कि क्यूबिट के बाद में और कौन कौन से कॉन्सेप्ट्स हैं, राइट? एंड देन वन डे दिस टॉपिक विल एग्जॉस्ट, तो फिर कुछ और पूछेंगे, है ना? बट बाय दैट टाइम न्यू टॉपिक्स विल कम अप. नो, डोंट वरी, यूपीएससी डोंट हैव एनी प्रॉब्लम विद द टॉपिक्स. नाउ व्हाट इज देयर दैट इफ वी टॉक अबाउट दिस एंटायर स्कीम, अगर पूरे स्कीम को देखें दैट हाउ दैट इज फंक्शन, तो लाइक आई टोल्ड यू दैट ई कोलाई, it's there in bacteria. So this is bacteria, let's say E. coli bacteria. And in this bacteria, this is DNA. This is what? DNA. This is the DNA of bacteria. And out of the DNA, this is called CRISPR. This is called CRISPR. This area is CRISPR, right? Now this bacteria is attacked by a virus. So virus has attacked this bacteria. And when the virus attacked this bacteria, the virus will pump its DNA. इसने अपना DNA क्या किया? अंदर डाल दिया. Virus DNA, this is viral DNA. This is virus. And viral DNA will attack over the entire system. It's like clash of 2 DNA. You get my point? Bacteria has got own DNA. But virus's DNA can be stronger enough, and virus DNA will take over the entire bacterial system, and then it's all gone, right? So only one way to destroy this infection to destroy this DNA. When I take regular foundation class, then in a very first day I talk about bacteria, about virus, right? और उस वहां पे मैं बताता हूं कि वायरस क्या है. तो वायरस इज लाइक टेररिस्ट. वायरस इज इक्वल टू टेररिस्ट. Virus don't have any cell structure, right? But virus needs cell for their survival, right? Like the way terrorist can enter into the India. If terrorist is entering in India, terrorist is not having any house, home, family member, nothing is there. But to survive here in India, terrorists need a setup. Terrorists need some structure. Terrorists need a house. तो टेररिस्ट क्या करता है? किसी घर में घुस जाता है. वेयर इम्यूनिटी इज नॉट स्ट्रॉन्ग इनफ, वेयर द डिफेंस सिस्टम इज नॉट स्ट्रॉन्ग इनफ, टेररिस्ट विल एंटर इनटू दैट हाउस. एंड टेररिस्ट विल हैव व्हाट? AK-47. So terrorist is having AK-47. So this virus DNA is what? AK-47. Now if the virus DNA is entering in our body, in our cell, we also have DNA. So how come our own DNA is not acting properly and the viruses DNA are dominating over it? ऐसा हो सकता है. तो आप सोच लो, टेररिस्ट इज हैविंग AK-47. AK-47 कितने केजी का होगा? 50-60 केजी का होगा. लेकिन हमारे घर में तो 200 केजी लोहा पड़ा हुआ है कि नहीं? 2 सिलेंडर पड़ा हुआ है, वही हो गया काफी. It doesn't mean that how much iron you have. It's just whether you have potent iron or not. Virus is having a small segment of the DNA, but the DNA or RNA of the virus is very dangerous. You got my point? In our body, in our cell, we have 30,000 genes. We have many more junk DNA. So in comparison with viral DNA, in our cell, the DNA amount is too high. But then our DNA is not able to compete with viral DNA. You get my point? And only those viruses are dangerous. ये भी समझ लो आप, है ना? आपके घर में टेररिस्ट उसी से डरोगे ना जो AK-47 लेके आएगा. कोई डंडा लेके टेररिस्ट घुस जाए तो क्या उससे डरेंगे आप? मारपीट के भगा देंगे. तो ऑल वायरसेस आर नॉट डेंजरस टू अस. किसी को वेरिएंट मैंने कहा था अभी क्लास के शुरू में, है ना, कि किसी का वेरिएंट ऐसा बना कि उसके पास AK-47 आ गया, अब वो ज्यादा डेंजरस है. एक और वेरिएंट बना, उसके पास तोप आ गया, कैनन, और ज्यादा डेंजरस हो गया. किसी का ऐसा था कि AK-47 गायब हो गया, अब क्या आ गया? स्टिक आ गया. नो प्रॉब्लम, अ लिटिल बिट ऑफ प्रॉब्लम विल बी देयर एंड विल सॉल्व इट. So now this is like AK-47 which entered and only one way to get rid of it, destroy it. तो क्या होता है? मैकेनिज्म क्या है कि दिस, दैट इज नेचुरल मैकेनिज्म है कि दिस E. coli will make gRNA, right? And this gRNA will have, यहां पे क्या होगा? दिस इज Cas9. What is that? Cas9. And now this gRNA will go and combine over here. ये जाके किधर कंबाइन करेगा? इधर. This is gRNA. gRNA will have instructions to recognize this viral DNA. And now this one, this Cas9 will cut it over here. है ना? Who will cut? Cas9. But who will take Cas9 to the viral DNA? gRNA. Guide RNA. Guide का काम क्या होता है? To take it to the place where it is required to go. You get my point? How does it happen? कैसे कर लेता है? So remember one thing. First of all, let's say you know that DNA has got adenine, guanine, cytosine, thymine. ये तो पता है. तो देखो यहाँ, अगर ये वायरस DNA है तो यहाँ पे एडेनिन, थाइमिन, एडेनिन, guanine, cytosine, right? These things are there. Guide RNA will have, ये guide RNA है, guide RNA will have complementary stand just opposite of it. You know that DNA में हम पढ़ते हैं कि एडेनिन डबल बॉन्ड थाइमिन और साइटोसिन ट्रिपल बॉन्ड गुआनिन. आरएनए में एडेनिन है तो यहाँ पे क्या होगा? यूरासिल, एडेनिन, यूरासिल, साइटोसिन, गुआनिन. It's just complementary. It's like lock and key. You get my point? It's like lock and key. That is why it is called guide RNA. So what does, एक बार ध्यान से सुन लो, देखो, what does it do? This DNA has got a CRISPR segment. CRISPR segment will form guide RNA, and guide RNA will form just complementary to the viral DNA. Viral DNA का complementary बनेगा. Somehow this E. coli or this bacteria will learn what is written onto this DNA. If this DNA has this information that has been read by bacteria, and now the bacteria has made what? gRNA. ये क्या है? Guide RNA. And guide RNA will have complementary information. It's like lock and key. And now this guide RNA is having what? It is having here, it is having what? Cas9. So now this guide RNA will not match anywhere, but it will match only with the viral DNA. It will go align itself with the viral DNA and Cas9 will cut it. इससे प्रिसाइज क्या हो सकता है? इससे प्रिसाइज कुछ हो ही नहीं सकता है. Right? So we got this information in 1987. 1987 में हमने पहली बार पता चला कि अच्छा वायरस के बैक्टीरिया के पास भी इम्यूनिटी सिस्टम है. एंड देन 2007-8 तक हमने इसको पढ़ के सब कुछ क्लियर कर लिया. 2012 में पहली बार ह्यूमन बीइंग के लिए यूज किया, एनिमल के लिए यूज किया. 2020 में नोबेल प्राइज मिल गया. एंड नाउ दिस इज कंसिडर्ड टू बी वन ऑफ द बेस्ट थिंग दैट बायोटेक्नोलॉजी कैन गिव अस. राइट? तो व्हाट वी आर गोइंग टू डू, हम लोग इस गाइड आरएनए और इस पूरे सिस्टम को निकाल लेंगे, है ना, और निकाल के इंफॉर्मेशन बदल देंगे. अभी इंफॉर्मेशन किसका लिखेंगे? कैंसर जीन का. यू गेट माय पॉइंट? So now you do just one thing. You take the system out, you change the address, you put it inside the cell. You get my point? अभी किसका एड्रेस लिखा हुआ है इसके ऊपर? वायरल के डीएनए का. चेंज तो कर सकते हैं, राइट? कभी कभी होता है ना साइंस फिक्शन में, कहानी की किताबों में, इनवेलप पार्सल बॉम्ब सुना है? पार्सल बॉम्ब. द पार्सल वुड कम, देयर विल बी अ एड्रेस, एंड इनसाइड द पार्सल देयर विल बी अ बॉम्ब. Because it's your address, if you open, the bomb will blast. It might create some problem. So it's a kind of parcel bomb only. In this parcel bomb, the address is written of virus, viral DNA. Now we took the parcel bomb out. उसका एड्रेस हटाया और दूसरा एड्रेस चिपका दिया और फिर अपने सेल में डाल दिया. अभी क्या है? वायरल डीएनए का एड्रेस हटाकर और हमने कैंसर जीन का डीएनए एड्रेस चिपकाया. And then we are using it same way. So imagine a situation that if we have access to this gRNA and Cas9, then we can remove this address, we can add the address according to our own wish, and then we can introduce it inside our cell, and now this system will eliminate or remove only those genes that has been instructed upon. है ना? So you can write guide RNA has complementary information of the target DNA, complementary information of the target DNA, right? And Cas9 can cut that target. Cas9 can cut the DNA. In this way, genome editing can be done. Genome editing can be done without much human interference. And with highest accuracy. Right. एक छोटा सा पॉइंट और है, आरएनए इंटरफेरेंस. right? So what we are going to do in RNA interference, we are going to interfere with the messenger RNA. So you can write in this technology, messenger RNA, messenger RNA is destroyed by another RNA molecule, introduced from outside, another RNA molecule introduced from outside, and this will stop protein synthesis. This will stop protein synthesis without eliminating the gene. Without eliminating the gene. राइट अभी सबसे बड़ा जो एरिया है जीन थेरेपी का करंट में, दैट इज कॉल्ड CAR-T सेल थेरेपी, राइट? यू नो दैट? हाँ जी, हाँ हाँ, अभी अभी उसी इसी के बाद दे रहा हूं. आई एम जस्ट इंट्रोड्यूसिंग द टॉपिक एंड देन. हाँ, मुझे बोला कि 10:30 में ब्रेक, राइट? हाँ ठीक है. मेरी आदत है ना, लाइक आई डोंट गिव ब्रेक. मैंने 40% में ब्रेक देने का मन नहीं करता. फिर वो अंदर लगता है कि अभी तो 40% ही क्लास हो, अभी 60% और लेना है. तो गिव इट एट 60-70% ना, कि लास्ट में कि अब तो एक ही घंटा बस है ना. तो हर इंसान का अलग अलग मोटिवेशन होता है ना. तो राइट. एंड यू नो दैट इंडिया हैज आल्सो डेवलप्ड इट्स ओन CAR-T सेल थेरेपी. दिस इज द बेस्ट थेरेपी दैट यू कैन हैव इट फॉर द कैंसर टुडे, है ना. लेकिन मैंने इसलिए बताया कि लेट्स इंट्रोड्यूस इट एंड गो फॉर द ब्रेक, और फिर मिलते हैं एंड देन विल टॉक मोर अबाउट इट. So after the break, we are going to complete this. That's the last topic of biotech, and then we'll move on to the nanotech. चलिए, हाफ एन आवर के ब्रेक के बाद मिलते हैं. So we'll talk about this CAR T cell therapy, right? And it's a gene therapy only, but nowadays for the cancer treatment, this therapy is being used. And recently, India has developed its own CAR T cell therapy. So first of all, so CAR, that is one word, CAR, that is called chimeric antigen receptor. So that is one. T cell, that is type of WBC, that is what the T cell. So we need to get the basic concept of it first. The whole idea is that T cell is a type of WBC which can kill, right? It's a killer cell, right? We know that in a basic concept we talk about, let's say, if I talk about WBC, so WBCs can be of 5 types, right? One is called eosinophil, then neutrophil, then there is basophil, then there is monocytes, and the last one is called lymphocytes, right? So these are the 5 types of WBCs, and these lymphocytes can be of 2 types. It can be B cell and it can be T cell, right? So B cell and T cell. This B cell is responsible for formation of antibody. So antibody is synthesis, right? And this is killer cell. Killer cell. I'll tell you the difference between both. But this is a basic concept of it that you should know before you get into the entire scheme. Right. So let's focus on lymphocyte, B cell and T cell. B cell produces antibody, and this antibody can kill. It's like, killing a pathogen by chemical, right? A pathogen, or let's say it's a bacteria is there, then this can kill, like this antibody can kill. That is why we take vaccines. Vaccines are taken because of this only. Because in vaccine, we compel our B cells to produce antibody. Let's say in a COVID vaccine, what we have taken? In COVID vaccine, we have taken the part of COVID virus, right? In a polio vaccine, what we put in? A part of polio virus. So we train our body. In any kind of vaccine, we are going to introduce some kind of pathogen in our body, right? That is the best way to get the vaccine, right? So let's say, polio drop, what we do, like in the next topic, we are going to talk about the vaccines also. So in polio drop, there is a polio virus. You put the polio virus in the body. Now, once the polio virus is there in the body, the polio virus will infect our body, because that's the weak polio virus. It's not a potent polio virus. It will not create polio, but it will create infection. So our WBC will fight back, and the B cell will produce antibody against the polio virus, and it's all done. Once antibody has produced, it is done, right? T cell is something which can get inside the cell and kill it, right? If directly being killed, it's a function of T cell. If it is being killed by a chemical produced, that is antibody, then it's a B cell. So most potent one is what? T cell. Now T cell can kill anything which is coming from outside. Right? It has got killing potential, very good killing potential if it is coming from outside. Any pathogen, anything which is coming from outside. Now scientists are willing to create, they wanted to change this T cell, that T cell will kill the cancer cell. You got my point? CAR T cell therapy is all about changing your T cell in such a way that your T cell will kill the cancer cell. Now, cancer cell is not coming from outside. Cancer cell is our body cell. Cancer cells belongs to the body system itself. Our own cell now dividing uncontrolled way, then it is called cancer cell. And now cancer cell, because it's our own cell, WBC will never kill it. Because WBC has got just one thing, self and non-self. Self don't kill, non-self kill it. But we can do something like we can train it. हम लोग ट्रेन कर सकते हैं ना उसको. Like our WBC is something like, our armed forces or police or security agencies that we have. It's like WBC. And we have already indicated all our security agencies and armed forces that you should be searching and killing and eliminating anything which is coming from outside. You should not attack on our own citizen. And for that, they have ID system. ID. Policemen are also having the same ID card. And we are also having same ID card. Right? So whenever you go somewhere, they will search for the ID. They will look at the ID and they will leave you. Because you belong to self. It's the same ID card there with the policeman. The same ID card belongs to us also. Right? But if some other ID card would be produced, then the policeman will take some action. You get my point? So action will be there only when the ID card is different. Only from something, somebody is coming from outside. लेकिन ऐसा भी तो होता है हमारी कंट्री में कुछ नक्सलिज्म and all kind of things will come up. It's like cancer only, right? Our own system is working against us, killing us from inside. In that way, police has tough time to recognize. आप देखते ही हो ना, नक्सल वाला जो एरिया होता है, you never know who is Naxalite and who is the normal man or women. Because in a daytime, it's like normal. And whenever it is, they have to plan some kind of attack, they will all become active. So in that way, you need to train police to recognize a particular group. And then you try to arrest it, eliminate it. So that is same thing that we are going to do. तो होता क्या है यहाँ पे? That in CAR T cell, one thing we should know that if we have a cancer cell, it's a cancer cell. So cancer cell will have, it will have some kind of special protein present over here. So that is antigen or ID of cancer cell. Right. So this is a special protein which is present over cancer cell. You got it? This protein will appear only when the cell is cancerous, nowhere else. Right? And then what we have done, we have taken the T cell out. We have taken the T cell out. So from our body, the T cell has been taken out. On this T cell, in this T cell, some gene has been added. And due to addition of gene, now this T cell will have a receptor. This is what? Receptor. This is called CAR, chimeric antigen receptor. You get my point? Now this antigen is protein. ये भी क्या है? It's a protein. It's protein. And this is also what? Protein. Protein. So the whole idea is take the T cell out of the body. Patients के body से T cell निकालो. Now you do one thing. Introduce a gene inside the T cell. Due to this gene, this T cell will have a receptor protein present on its cell membrane. And now this receptor will be what? Complementary to the cancer antigen. It will go and catch the cancer cell and it will kill it. Recognition point. They don't have any receptor to recognize this. They have receptor for everything else that can come from the outside. But it doesn't have receptor for our own cancer cell. So what we need to give? We need to give a kind of receptor, a gene. So one gene that is there And that gene will do what? It will create a special receptor onto the T cell, and now this T cell will recognize the cancer cell. You get my point? So just by adding a gene, you are putting something onto the T cell, and the T cell is trained enough. It is, being trained to recognize the cancer cell. So you can write in CAR T cell therapy, in CAR T cell therapy, in CAR T cell therapy, T cell of the patient, T cell of patient, T cell of a patient is taken out and genetically modified, and genetically modified and genetically modified. Full stop. Genetically modified T cell, genetically modified T cell will develop chimeric antigen receptor, chimeric antigen receptor, chimeric antigen receptor, and this receptor will recognize the antigen or surface protein. This receptor will recognize the antigen or surface protein present on cancer cell. Present on cancer cell. Right. Next point. This modified T cells will be introduced in the patient body where it will detect and kill. It will detect and kill cancer cell. It will detect and kill cancer cell. Right. So this is what the CAR T cell. So this antigen receptor, that is called chimeric antigen receptor, and this is called antigen. It is antigen receptor, and this is antigen. So antigen receptor is going to recept the antigen, and cancer will have some antigen or surface protein. If our cell goes cancerous, it is bound to have a surface protein or antigen. But our immune system don't have any way to recognize it. So by gene therapy, we are giving a way to recognize it. So one gene introduction, and then it has been done. Recently, it's been in news because it's a very novel way to, treat cancer. But now India has developed its own way. That is called Next CAR19. So Next CAR19. Next CAR19. So you can write, it is a type of CAR T cell therapy. Type of CAR T cell therapy. CAR T cell therapy developed by developed by Indian company Immunoact and IIT Bombay. Immunoact and IIT Bombay. You're right. In this technology, these cells are designed. These cells are genetically modified. These cells are genetically modified and designed. These cells are genetically modified and designed to target cancer cell. Target cancer cell having CD19 surface protein, CD19 surface protein or antigen. CD19 surface protein or antigen. Protein name is not important, but that is how the 19 came. CD19 surface protein and antigen. So what is the difference between our own technology and CRT international technology? That's the price band. Price band has gone down now, right? So Indian CRT cell technology will be available in around 15 to 20 lakh rupees, still lakh. But those CRT cell therapy which is coming from the other country, that will be more than 40 lakh, right? So you can write one more point. Indian next CR19 therapy, is cheaper than the alternatives, right? So this is another example of gene therapy that we started talking about. Now, one more term that we'll talk about. You know that there is something called cellular agriculture. Nowadays, it's also making a lot of news around cellular agriculture. So cellular agriculture means that you use cell or living organism to create animal products. Animal products means that you can do like meat without killing the animals. Like cell, we have a stem cell, you know that. The stem cell can keep on dividing, it can produce tissues and organs. Similarly, now we have devised a way in which we don't need to kill animals, but still we can get the meat out of it. So cell will be taken into the lab. In the lab, it will be treated in a specific way, and then the cell division will occur, and it will give you lab cultured meat. Two benefits: one, ethical benefit that you don't kill animals, and second, the carbon footprint of this is less. Right? You know that animal husbandry has a lot of carbon footprint, है ना? तो उसका भी कम कर लेंगे. And not only that, cellular agriculture has the other way also. Like now the scientists have developed that there are certain kind of fungi. In that fungi, if we do precision fermentation, that fungal cell can produce milk and egg protein like albumin, right? Milk का मतलब वो एकदम वैसा ही वाला milk नहीं. Nutritional value will be like milk only, and the egg protein also. So you can write cellular agriculture is a lab-based process. Lab-based process of cell culture, to produce animal products, animal-based products. Animal-based product. Right, animal-based product. Next you can write, it is sustainable and ethical alternative. Sustainable and ethical alternative to traditional animal husbandry. Traditional animal husbandry. Right. Next you can write, carbon emissions can be reduced through this method. Right. Next you can write, cellular agriculture has two main methods. Cellular agriculture has two main methods. One is called cell cultivation. And when we do self cultivation, then it is like use of a stem cell and that will lead to meat production. Right. Then another is called precision fermentation. That is also part of it, where fungi will be used, use of fungi. And that will produce milk production and egg white production, egg protein production. Right. Egg protein production. Got it? So that's the concept of cellular agriculture. Meat and other animal product like milk and egg protein. एक और टर्म आता है ना जिसको कहते हैं सेल कल्चर्ड मीट, है ना? दैट इज अनदर टर्म दैट इज कॉल्ड सेल कल्चर्ड मीट. And Singapore became the first country in the world to allow the cell cultured meat to be sold in market. So if you go to Singapore, you might have some burger or hot dog, and that meat which is present inside will not be coming from the killed animal. Rather, Singapore has done a lot of experiment in that. उन्होंने 3D printing वगैरह से भी मीट बना के खिला दिया लोगों को एक्सपेरिमेंटल बेसिस पे. बट फर्स्ट टाइम सिंगापुर has now rolled out that it will be allowed, right? So next topic in that is called cell cultured meat. Cell cultured meat, right? Another name is called lab grown meat, cultivated meat. It is called cultivated meat or lab grown meat, right? So you can write, it is grown in the laboratory. It is grown in the laboratory using living cells or fertilized egg, or fertilized egg. Right. Next you can write, such meat can be customized to make more healthy, by adding additional nutrients. By adding additional nutrient. Right. Next you can write, other benefits of cell cultured meat. तो फर्स्ट क्या है? एथिकल कंसर्न, नो किलिंग, तो नो किलिंग. सेकेंड इज लेस कॉन्टामिनेशन, less antibiotic, and less environmental impact. Less environmental impact. Less environmental impact. Right? So that is about this biotech issue that we wanted to cover some of them. Now we'll move on to area called nanotech. And in nanotech, they are asking questions from nanotechnology also in prelims examination. 4 or 5 years back, they started asking, right? They asked question about fuel cell technology and other thing. But if you look into the, I mean, nanotech is an entire chapter, but if you look into the important aspect of it, the nanotech application in terms of human health is important, then energy is important, and agriculture is important. Three area is very important. Although again I'll advise you that three area that is important one, it doesn't mean that other area is not important. It's just like we are focusing on three areas, right? I mean, there are area like environment, defense, security, textile, electronics. These all are important too, right? So health is the primary sector that is going to get the first benefit of the nanotechnology, because health is the biggest area of nanotechnology. Energy is like that only, and agriculture, because you know that India became the first country to roll out nano urea liquid, right? So here we have nano urea liquid, that is the topic which made this entire area important. Rather, we'll talk about nano urea liquid also, and then we'll talk about the other aspect of agriculture as well. When we talk about energy, then it's fuel cell and hydrogen fuel. That make the entire thing very interesting and important, right? And in the health sector, targeted drug delivery to cancer cell for cancer treatment, right? For cancer treatment, that become important. So we are recognizing three areas with three different examples. So we should know that why these areas are important, and one by one we'll take it, right? Right now, we just talked about targeting cancer cell with the help of T cell therapy, right? T cell therapy is biotech because we are, you know, putting some gene inside, then protein is being formed. It's like DNA to RNA, RNA to protein. So that is why it is called biotech. But a similar kind of thing can be achieved by nanotechnology also, right? In nanotechnology, let's say if we talk about health issue, health, or let's say nanomedicine, or we can call it nanobiotech, it's all same. You should not be getting confused with them. So health issues, nanomedicine, nanobiotech, or let's say nanobiosensor, it's all same. And the whole concept is that, like I told you, that if a cell is cancerous, it's a cancer cell. So cancer cell will have a special protein present on it. That is called surface protein of the cancer. You know that the same concept we are going to repeat because obviously concept will not change. Concept is same. Cancer cell has the same surface protein. It's the way to detect that will change. Right. So earlier, just when we was talking about T cell, we created a kind of a special antigen receptor onto the T cell which will catch over the surface protein. So that is what it's a surface protein. Let's not use the term antigen over here because the moment you use antigen, then it's moved towards biology. So it's a surface protein. And now in nanotechnology, we have developed a kind of receptor molecule. You can see like this receptor molecule is more or less like this only, lock and key mechanism where the things will function. This is your nano biosensor. And this nanobiosensor is very complementary to the surface protein. It will go and attach with it, right? And the moment it attaches with it, it will detect the cancer cell. So it's something which can detect the cancer cell. Now the thing is that how we got this? We have constructed it in lab. Because nanotechnology is all about constructing the things by assembling atoms and molecules together, है ना? तो nanotech में एक method है जिसको कहते हैं bottom up method. What we call it? Bottom up method. It's a nanotechnology method. In this bottom up method, what we do? We have atoms. Atoms को assemble करते हैं तो we get what? Molecules. And then molecules को assemble करते हैं, then we get the nanostructure. So we got something like this nanostructure. Nanostructure. You got my point? So that's the same nanostructure. Yes, please. No, it doesn't require. It's just independent. It doesn't require any cell structure. It's just independent because we have created by chemical mean. It's a chemical synthesis which has been done. Like the way we do chemical reaction, atoms to molecules, but molecules to nanostructure. Nanostructure means 10 to power minus 9. है ना? Cell का size क्या होता है? Micro, 10 to power minus 6, 10 to power minus 7. And inside the cell, it's 10 to power minus 9. So very tiny molecule you are going to make. Something which is very tiny, that is in nanoscale. Cell is at micro scale, 10 to power minus 6. And inside the cell, if there is a protein structure, if there is a mitochondria, lysosome, so obviously it will be smaller than cell. So below the micro scale, there will be which scale? Nanoscale. And in nanotechnology, we are operating at nanoscale. We are able to manipulate at nanoscale. So by the bottom-up method, we have taken few atoms, we have created some molecules, and then we have assembled some molecules to get the nanostructure. These nanostructures are independent one. It can go and attach with the cancer cell and it can give information outside. It is the entire system which can give information to you that in somebody's body, cancer cells have been detected. Rather, this can detect a cancer cell at very early stage. Because normally tumor will be visible, then only we'll able to get that this is cancer. But this will detect even if the cancerous growth is few cells only. Because we'll inject this nanosensor in our bloodstream. This will be so small that it can be injected inside the body. It will keep on moving inside the bloodstream. And the moment it come across any cancerous cell, it will go and attach. It will never attach anywhere else. It will always attach with the cancer cell. And all you need to do, you need to test your blood every Sunday, or maybe every day, maybe every month. Like the diabetic patient, they test their blood. खाना खाने के बाद एक बार टेस्ट कर लेते हैं कि ब्लड शुगर क्या है. It's so easy. The sensor is already there in your body, moving inside the blood, detecting the cancer cell. And as a precautionary measure, if the person is at high risk of cancer, then person need to take a small drop of blood out of the finger, put it onto the paper strip, put it inside the machine and get the reading. Like the way people are getting reading of their blood sugar in their house only. You get my point? So we are moving towards that. It's a home testing kit of cancer. वो भी possible हो जाएगा. हो चुका है basically. बस price band बहुत ज़्यादा है. That's why it's not getting popular. एक तो ये हो गया. This is what cancer detection. And detection sensor is already there in our body. But now scientists are thinking, rather they thought that why only detection? If we are able to detect cancer so precisely, ये cancer ही है, because this surface protein can come up only with the cancer, and this will attach only when it finds the cancer protein. That means if it detects, then 100% surety is there that cancer is there. है कि नहीं? तो do one thing, एक और काम करते हैं. So with this nano sensor, we are going to add a nanostructure. This is also a nanostructure. It's a drug carrier molecule. What is that? It's a drug carrier nanostructure. Example के रूप में लिख सकते हैं fullerene. You know fullerene? It's a carbon allotrope. And inside fullerene, we are going to add some drug. इसमें क्या डाल दिया? Drug. It's cancer drug. Now, what we got is basically targeted drug delivery method. है ना? Targeted drug delivery method. Now you see, this nanosensor will attach to surface protein of the cancer. It means this nanosensor is going to attach only with cancer cell. And moment is attached, it will release the drug on the cancer cell. So cancer cell will not only be detected, but it will be killed also. This drug is locked inside. It never gets released unless it attached with the cancer cell. And this entire scheme is known as what? Targeted drug delivery approach. And that is the biggest thing that we are going to see in nanotechnology, in the next big thing in the medicine sector. Right? तो पहला फेज क्या आएगा? पहले फेज में सिर्फ इतना आएगा. साइंटिस्ट हैव डेवलप्ड इट. इतना छोड़ दो ये वाला पार्ट. और अगर हम सिर्फ इसकी बात करते हैं तो दैट इज कॉल्ड हाईली सेंसिटिव डिजीज डिटेक्टर. व्हाट वी कॉल इट? हाईली सेंसिटिव disease detector. And with highly sensitive disease detector, if you add drug carrier molecule, then the entire system will become what? Targeted drug delivery system, right? So you can have it in two parts now. So if we talk about, so this whole part, अगर पूरी की पूरी बात करें, so that is what? That is targeted drug delivery method. And we talk about just this one, right? If we talk about just this one, this white one, so that is what? Highly sensitive disease detector. Got it? So, I mean, nanotech, they won't ask anything because it's a more concept. Nanotechnology is a future era of technology. It's not present day technology. Majority of the nanotechnology approaches would come up in future. It's there in lab. Lab, they developed it. They have tested it also. But we have some problem with that. The problem is that why to unnecessarily put something into the bloodstream? Hai you are putting inside the human body. Even if you get some, I mean, many people still not got the COVID vaccine. अभी भी मिल जाएंगे बहुत सारे लोग जिन्होंने कहा कि नहीं I won't take COVID vaccine. Why? Because how can I take a vaccine which has been developed in just 1.5 years? इसका मतलब है कि प्रॉपर ह्यूमन ट्रायल नहीं हुए और रियलिटी भी यही है. सेकेंड ट्रायल के बाद इंडिया में भेज दिया था, जबकि मिनिमम कितने ट्रायल होने चाहिए? 3. तो मेनी वैक्सीन हैव बीन रोल्ड आउट इन इंडिया आफ्टर 2 ट्रायल्स बिकॉज सेकंड ट्रायल में बहुत अच्छा रिजल्ट आया. बहुत अच्छा रिजल्ट आया, किसने देखा? सवाल तो ये भी है ना. आई मीन बहुत अच्छा बहुत बुरा, इट्स अ वेरी रिलेटिव टर्म. इट्स ऑल डिपेंड्स अपॉन दैट. ऑब्वियसली गवर्नमेंट भी लैग्स कर गई कि ठीक है 2 पे रिलीज करो. सीरम इंडिया ने भी कहा कि ठीक है वी हैव गॉट अ वेरी गुड रिजल्ट. नाउ देयर इज अ वेरी गुड रिजल्ट. दिस वेरी गुड इज वेरी रिलेटिव, है ना. तो मेनी पीपल आर देयर इन इंडिया हु रिफ्यूज टू गेट द वैक्सीन. मैंने कहा कि ठीक है हम ट्रेवल नहीं करेंगे, वैक्सीन नहीं लेंगे. राइट. एक हमारे फ्रेंड है, ही हैज नॉट टेकन वैक्सीन. ही यूज्ड टू टीच पॉलिटी, आई मीन अलोंग विद मी. तो when we all were going to take the vaccine, he was like, I will take a Sputnik. I'm waiting for that. And नहीं लिया. And now everything nowadays, you know that heart attack हो रहे हैं और लोग अब उसको भी जोड़ देते हैं किससे? कोविड से. कि वैक्सीन लिया था ना, अब वही सब इंपैक्ट हो रहा है. एंड दिस पर्सन एवरी टाइम ही विल कम, ठीक है ना, हेल्थ वेल्थ वैक्सीन लिया ना आपने? हमने हाँ लिया हुआ है. सो द आई मीन आई एम नॉट सेइंग दैट दिस पर्सन इज राइट और वी आर रॉन्ग हु हैव टेकन इट. लेकिन हाँ एक तो है ना, दिस क्वेश्चन इज देयर दैट व्हाई टू टेक दिस मॉलिक्यूल इनसाइड द बॉडी if there is no proper and extensive study has been conducted, है ना. And second thing, price band is very high. It's very costly. So that is why nanotechnology is holding all the possibility for the future. Because there is one problem with that. And problem is that these molecules are so tiny and so small that let's say if I feel that I'm at high risk of cancer and I inject this targeted drug delivery system in my body. So one thing is there, the possibility is very high that I'll be cancer proof. The moment cancer cell will occur and appear in my body, it will go and attack and kill it. Lekin at the same time, these are so tiny molecule that it can interfere with the other organs, other process, other physiological activities. And there has been some kind of cases in experiment where some nanoparticles entered into the brain of animals and then it has created some problem out there. जिसको हम साइड इफेक्ट कहते हैं. Although targeted drug delivery system has a biggest benefit that side effect will not be there, है ना. Because in cancer treatment, if you look into, then cancer treatment has got of lots and lots of side effect. Whether it is chemotherapy or radiotherapy, में क्या होता है? If a person is going through cancer treatment, then side effects are very severe. Hair loss, skin become black, person lose a lot of weight, because these chemicals will not only kill cancer cell, but it will kill cancer cell as well as healthy cell. Normally that we are doing right now. But this will only kill cancers. So one of the biggest benefit of targeted drug delivery system is that side effects are very less. But then they also talk about what will happen if this entire system is interfering with the other system of the body. Then it might create what? Severe side effects. That is why we are not using it. You can write one example. Cornell University, USA. Cornell University, USA has developed a nanomachine, which can selectively kill cancer cell in bloodstream. Cancer cell in bloodstream. Cancer cell in bloodstream. In bloodstream, blood flow, right? Next you can write DRDO along with IISc Bangalore. DRDO along with IISc Bangalore has developed typhoid detection kit. Based on nanotechnology, nanosensors, right? So this is the most prominent area. Now the second area that we are going to discuss is energy and environment both, right? Because energy can never be complete unless we talk about it in terms of environment. So energy, and obviously it has got some thing to do with the environment, right? Why? Because we always look for clean energy avenues, right? With lowest carbon emission, or best would be there is no carbon emission. That is why hydrogen fuel is being explored, because hydrogen fuel will not have carbon emission, right? Hydrogen will burn with oxygen, will produce H2O. And then that is why we are focusing more onto the solar energy, because solar energy will not have carbon emission. I mean, two energy sources that we are focusing more at the present time is solar and hydrogen, है ना? बहुत ज़्यादा कर रहे हैं. उसके अलावा भी है, चलो, bioethanol and all kind of thing. But these two has got a lot of potential. And nanotechnology is helping in both of them. Nanotech says that with integration of solar cell and hydrogen fuel, we can create a system where 24/7 energy would be available without any carbon emission, right? And for that, what they are going to do, so one is nano solar cell. Now you have to listen very carefully so that you can catch all the thing, and you can write also nano solar cell. So there is one solar cell that we are using right now. Abhi to use kari na solar cell. That solar cell that we use right now at the present scenario, that is called silicon solar cell, isn't it? It's a silicon solar cell. Good enough. No problem with that. Silicon solar cell are very good. लेकिन वेरी गुड का मतलब है कि इससे ऊपर भी तो कुछ हो ही सकता है. तो नैनो सोलर cell will be better than silicon solar cell, first of all, right? So silicon solar cell is not bad. It's very, very good. But even the superlative would come up, and that would be known as nano solar cell. Now this nano solar cell will be made up of what? It will be made up of graphene, that is a nanomaterial, plus CNT. Graphene and CNT. It's a carbon nanomaterial. So one thing is there, that nano solar cell will be made up of graphene and CNT. If you talk about silicon solar cell can trap only one spectrum of the sunlight. And that is what? Visible spectrum. That is a light. But solar radiation do not have only light. Solar radiation have three spectrum: light, ultraviolet, infrared. है ना? But silicon solar cell, out of these three, will be able to harness energy only from one. That is called light. But this will harness energy from two. है ना। तो ये एनर्जी किससे किससे ले रहा है, देखो यहां से, दिस इज विजिबल लाइट, विजिबल स्पेक्ट्रम VS, एंड देन इंफ्रारेड IR. So if a solar cell designed with graphene and CNT is able to harness energy from visible spectrum and infrared both, then energy potential will be more, है ना, ज्यादा होगा ना. So what will happen, that as output here you can see like energy will be more energy will be produced, right? So you should be very clear about it. It's there in diagram only. Clear hai explanation. If they say nano solar cell kisse banega, graphene CNT. Why it will be more? Why it will be having more energy potential? Because it will trap solar light and infrared radiation. Moreover, you know one thing: this graphene and CNT is best conductor. It's best, I mean, 1,000 times better than silver, right? So if a system is highly conducive, it means resistance is low, right? And you know that resistance is not good for any system. More resistance, more energy will be eaten up to overcome the resistance, and output will be compromised, isn't it? You know that your mobile phone sometime it become hot. कभी होता है ना कि गर्म हो जाता है. Or the moment your mobile becoming hot, you will realize that your battery has gone down drastically. 20 minutes में battery 80% gap. Because this heat is coming out of resistance. There are some problem. That is why internal resistance of the mobile has gone up, and lots of electrical energy is now converting into heat energy. That is why your mobile energy efficiency has gone down. So silicon solar cell, they have internal resistance because there we use silver wire to connect two cells. Why do we use silver wire? Because we know that silver is a least resistor. लेकिन कुछ तो resistance है ना. ये graphene और CNT silver से 1,000 times कम resistant ऑफ करता है. That means 1,000 times more conductor है. So it means that it will not eat energy as much the silver or normal solar cell is eating. So output will be increased once again. है ना. एक और बात आप जानते हो ना, लेट्स से ये सोलर पैनल है, सिलिकॉन सोलर पैनल, राइट. इन सिलिकॉन सोलर पैनल वी पुट सिलिकॉन सोलर सेल्स लाइक दिस. दीज आर सेल्स, ये सेल्स है. देयर आर 12 सेल्स. लेट्स से इफ आई एम टॉकिंग अबाउट 12 सेल्स, एंड ऑल 12 सेल्स आर कनेक्टेड टुगेदर विद द हेल्प ऑफ सिल्वर वायर. तो इंटायर बोर्ड, ठीक है, हम लोग आज बहुत एफिशिएंट सोलर सेल बनाते हैं. If you look at the board, you will find that almost entire board is solar cell. But there are different cells. And all different cells are connected together by the silver wiring. I mean, you cannot say that if this is this surface area, then 100% surface area can never be solar cell. You get my point? It will be 90%, 80% only. But graphene is a highly conducive, highly conductor. And graphene is the entire surface. You got my point? It's an entire surface. There is no interconnection needed. It's a conductor itself. So in a graphene solar cell, it won't be 90-30%, 90%, 80%. It will be entire surface of the graphene. That will be solar cell. And graphene is so easy to use because you need very thin layer of it. That you can paint it over a wall. You can spread it like a roof, I mean bed sheet. So high surface area, right? So energy output will be even more because of the following regions I am writing over here. तो यहाँ पे energy output ज़्यादा क्यों होगा? Because of surface area, more resistance, less. You get my point? So all the thing will make this highly energy efficient. Surface area more and resistance will be low. इधर क्या है? Energy harvesting from two different spectrum. All the thing will make it better than the normal solar cell that we have. And energy produced by nano solar cell will be much more than the normal solar cell. And only a part of it we are going to use it. हम सब यूज शायद कर भी नहीं पाए, और ऐसे भी करेंगे तो कुछ ना कुछ करेंगे. सो वी कैन डू लाइक दिस. हम लोग इसमें एक और बना सकते हैं कि लेट्स से यूज इन डे टाइम. डे टाइम में तो लाइट है ना, सनलाइट, लेकिन अभी सरप्लस एनर्जी होगा. इधर क्या होगा? सरप्लस एनर्जी. So surplus energy will be there. This surplus energy will be given to fuel cell or gas cell. There is another concept. What we call it? Fuel cell or gas cell. So we are going to provide this surplus energy to fuel cell or gas cell. That is being transferred in daytime. क्योंकि daytime में तो काम करेगा ये. If the sun goes down, it's all gone. But we need energy at the night also, right? And for that, we are going to store this energy somewhere. And we are going to store this energy in fuel cell. So let's say, talk about fuel cell. What is fuel cell? Fuel cell is a platinum-based nanostructures that we have developed. And this fuel cell, you might have heard about. UPSC ने क्वेश्चन पूछा हुआ है फ्यूल सेल पे. फर्स्ट क्वेश्चन ऑफ नैनो टेक्नोलॉजी, आई मीन ग्राफीन पे पूछा था एक बार, फिर यही पूछा. तो फ्यूल सेल इज प्लैटिनम बेस्ड स्ट्रक्चर दैट विल स्प्लिट वाटर मॉलिक्यूल इनटू हाइड्रोजन एंड ऑक्सीजन. यू नो हाइड्रोजन इज फ्यूल, है ना? तो व्हेनेवर दे से फ्यूल सेल एंड गैस सेल, इट्स ऑल अबाउट हाइड्रोजन फ्यूल. कभी आपने न्यूज में पढ़ते हैं ना कि BMW और Mercedes they have made a car which is running on a fuel cell and hydrogen, है ना? हालांकि वो कार बाजार में बिक नहीं रहा अभी. इट्स अ प्रोटोटाइप, इट्स अ कॉन्सेप्ट कार. बट whenever this, I mean automobile company would come up with a hydrogen fuel and concept car, then they will always come with one term that is called fuel cell, right? Although fuel cell at present time is very costly, it's not cost effective, but ठीक है, कोई दिक्कत नहीं. इट विल कम चीपर, राइट? देखते देखते देखो कितना चीपर हो गया. पहले एलसीडी एलईडी कितना महंगा आता था. आई मीन स्मॉल एलईडी 30-inch टेलीविजन, देयर वाज अ टाइम व्हेन इट केम टू द इंडियन मार्केट, इट वाज 1 lakh rupees, and now you get in 10,000 rupees also. So that is how technology become cheaper over the period of time. But here you need to understand, fuel cell and gas cell, it's the same thing. With the help of platinum catalyst. Platinum किसकी तरह काम कर रहा है यहाँ पे? Catalyst. ये catalyst. And then it will break the water molecule into hydrogen and oxygen, and it will store it. And for this breaking, energy कहाँ से आ रही है? Right? Now you store it. Keep on storing the hydrogen in that. If you wish, you can eliminate oxygen. No problem. Oxygen की दिक्कत नहीं है हमारे पास. Oxygen is there in atmosphere, but hydrogen will keep it safe. And when this fuels, I mean this solar cell stops working, ये stop काम करना बंद कर दिया. Now we can take advantage of this. अभी क्या करेंगे? रात में इसको बर्न करेंगे ऑक्सीजन के हेल्प से. क्या होगा यहाँ पे? ऑक्सीडेशन ऑफ हाइड्रोजन, and that will give you what? Energy. At night. आप डे में भी यूज करो, मैं तो कॉन्सेप्चुअल बात कर रहा हूं. एनर्जी एट नाइट. अब आप देख लो, इट हैज गिवन यू एनर्जी बैक विद ऑक्सीडेशन ऑफ हाइड्रोजन, आई मीन हाइड्रोजन विद द हेल्प ऑफ ऑक्सीजन, और वापस क्या बन गया? वाटर, राइट? सो डे टाइम यू गेट एनर्जी हियर, नाइट टाइम यू कैन गेट एनर्जी हियर. इन दिस एंटायर प्रोसेस, इज देयर एनी ग्रीन हाउस गैस इमिशन? फॉर दैट मैटर एनी गैस इमिशन? आप चाहो तो ऑक्सीजन को भी क्यों निकालना? नहीं निकालो. So there the future lies, right? Now these both concepts can be differentiated. अगर अलग अलग बात करो तो नैनो सोलर सेल, एनर्जी, फ्यूल सेल, हाइड्रोजन फ्यूल. You get my point? UPSC has asked question in prelims examination, and it was like, which of the following statement is true regarding fuel cell technology? The first option was fuel cell has platinum nanoparticle as catalyst. So true. Second, it splits water molecule into hydrogen and oxygen. That is also true. And third one, that fuel cell can produce AC current. That is wrong because no cell can produce AC current. You get my point? So UPSC will not make something which is easy for you. उसको पता था कि भाई नैनोटेक की क्लास हुई होगी तो फ्यूल सेल तो पक्का ही पढ़ा होगा, लेकिन फिजिक्स में कहीं पढ़ा होगा एसी डीसी, सेल बैटरी. तो लेट्स कन्वर्स दिस टू थिंग टुगेदर एंड टेस्ट द प्रेजेंस ऑफ माइंड ऑफ द कैंडिडेट. कि थर्ड ऑप्शन तक आते आते क्या वो अपने प्रेजेंस ऑफ माइंड को यूज कर पाता है कि सेल मींस डीसी, सेल नेवर मींस एसी, राइट? So after the examination, two kind of people will meet, two teacher. One is happy one, जो कहेगा कि sir I could connect it because I have read AC and DC. The second category of the people, they will come cribbing. Sir दो तो आपने बताया, तीसरा तो बताया नहीं आपने, AC या DC. तो उसका कुछ नहीं हो सकता, है ना? तो that is how we always try to make the things, cohesive. I mean, UPSC will go to any extent to make the thing difficult. Not difficult. UPSC is not making things difficult. UPSC को भी पता है कि 20 तो पन्ना नहीं है इससे, तो 20 तो ऐसे क्वेश्चन डालेंगे जो नहीं बनेगा. तो 75, 80. I mean, maximum you can attempt is 80, और minimum you can attempt is 70. Minimum का कोई नहीं है, लेकिन 70 तक तो चला ही जाता है इंसान. The moment you go beyond 80, your chances are now diminishing because now you will attract more negative marking. One or two numbers से जिनका भी छूटता है ना, they are not because they could not solve one or two question, just because they attempted more, right? तो जो the boundary line failure in prelims is not because lack of the knowledge, it is due to lack of the management. वो टेस्ट सीरीज का यही तो कॉन्सेप्ट है. Why do you need to do test series? And test series doesn't mean that I'm promoting test series that you go and buy it. No, I'm very student friendly teacher. I always say test series करना है ना, go to the bookshop, get the test series from there, photocopy 20 rupees में मिल जाता है, घर पे आओ बनाओ, है कि नहीं? If you can afford, you can go with that, कोई दिक्कत नहीं है ना. लेकिन टेस्ट सीरीज बनाते समय बनाओ जरूर उसको. And every time you go the test series in how, I mean घर पे बैठ के भी, तो just one thing you need to do, एग्जामिनेशन का जो इतना टाइम होता है ना और जो उसका डिसिप्लिन होता है उसको मेंटेन कर. इट्स नॉट लाइक 10 अभी बना लेते हैं फिर घूम के आएंगे फिर 10 बनेंगे, नहीं. लर्न हाउ टू मैनेज योर टाइम, दैट वी हैव 2.5 hours और 2 hours, जितना भी टाइम आपके पास है उसमें फर्स्ट 30 मिनट हाउ डू वी यूज इट, नेक्स्ट 30 मिनट हाउ डू वी यूज इट. व्हाट इज, जैसे क्रिकेटर क्रिकेट खेलता है तो स्ट्राइक रेट मेंटेन करता है ना, सो व्हाट इज माई स्ट्राइक रेट, हाउ कैन आई रिविजिट द क्वेश्चन. इफ देयर इज 1 question or 5 question that I have ticked कि इसको मैं रिविजिट करूंगा only when my total score is not up to mark. So are you able to get the time back to revisit it? You get my point? Because लास्ट में कई बार ऐसा होता है ना कि लास्ट का 10 क्वेश्चन पढ़ भी नहीं पाते लोग. There are times when people are not able to read the last 10 questions. और फिर घर आते तो पता चलता है कि लास्ट 10 क्वेश्चन में 8 आ रहा था उन्हें, है कि नहीं? तो it's a kind of time management. For that also you should go with that. Anyway, and normal cell means that, I mean, the battery that we have in our house, right? तो देखो उसमें तो इलेक्ट्रोलाइट वाला मामला है. इट्स chemical things are going on, but there we are having hydrogen. It's a fuel, will burn this. जैसे कि आप जेनसेट में क्या चलाते हैं, पेट्रोल, और पेट्रोल से क्या चलाते हैं फिर आप, इंजन, इंजन से क्या चलाते हैं, डायनेमो, electromagnetic induction and energy will come out. The same replace petrol with hydrogen. You get my point? So at the place of petrol, it's not like that fuel cell will directly. Fuel cell will store hydrogen. It will give you energy. And then with the help of that energy, you can also rotate the turbine or dynamo or electromagnetic induction. You can get energy out of it. तो वैसे निकाल लेंगे. And by the way, the battery you are saying, na, that battery is not something where you can store energy. Because battery, the moment you charge, the next moment is a start discharging. You get my point? इसमें क्या है ना कि एक बार आपने हाइड्रोजन फ्यूल इसमें फिल कर दिया, तो आफ्टर 1 year ऑल्सो इफ यू कम, यू विल गेट द सेम अमाउंट ऑफ हाइड्रोजन. यू गेट माई पॉइंट? दैट मीन्स एनर्जी इज स्टोर्ड, रियल सेंस में स्टोर्ड है. लेकिन अगर मैं अपने मोबाइल फोन की बैटरी को चार्ज करके फिर महीने भर मोबाइल फोन छोड़ दूं ऑफ करके भी, तो एम आई सपोज टू गेट द सेम अमाउंट ऑफ चार्ज और इलेक्ट्रिसिटी दैट आई हैव स्टोर्ड? नो, इट विल डिसिपेट. दैट इज व्हाई नाउ साइंटिस्ट हैव ऑलरेडी गॉन अवे फ्रॉम द कॉन्सेप्ट कि हम बैटरी रखेंगे और बैटरी में स्टोर करेंगे. वो तो टेम्परेरी मामला है. जैसे आपके इन्वर्टर में बैटरी लगे होते हैं ना घर पे, वी ऑलवेज कीप ऑन चार्जिंग. कभी ऐसा होता है कि अच्छा चार्ज हो गया बंद कर देते हैं? नहीं, बिकॉज इफ यू स्विच इट ऑफ देन आफ्टर सर्टेन टाइम इट विल ऑल गॉन, है ना. अब देखो इसमें दो मामला है, दैट यू गेट एनर्जी विदाउट ग्रीन हाउस गैस इमिशन, राइट. सो दैट इज हाउ वी हैव डन इट. एंड it's not like that any single country or single scientist have done it. No, this technology is very much known to everyone. In India also the things are going on. The only drawback of this technology is these things. ये क्या है टेक्नोलॉजी में? High cost. These things are very very costly. But then things will come up, है ना. I always say that any energy, कोई भी एनर्जी सोर्स कब बढ़िया माना जाएगा? लाइक एनर्जी और फ्यूल विल बी कंसिडर्ड बेस्ट विद 4 verticals, 4 characters. One is that high calorific value. That is one thing. Second one is low pollution. Low pollution. Third is safe transport and storage. And fourth, economical. Now, if you talk about fuel cell, 3 is already done. है ना. हाइड्रोजन के साथ एक ही दिक्कत थी, 2 दिक्कत थी. एक तो हाइड्रोजन के साथ सबसे बड़ी बात क्या है कि कैलोरिफिक वैल्यू हाईएस्ट है, इससे बेहतर किसी का हो नहीं सकता. Among all the fuel that you have, hydrogen will have the highest calorific value. It's proven. Second, हाइड्रोजन में पोल्यूशन नहीं है. हाइड्रोजन बर्न, you will get distilled water. No problem with that. पानी से क्या प्रॉब्लम हो सकती है? The only problem we had with hydrogen is safe transportation and storage was not there. Hydrogen explosive है, explode करता है. But fuel cell has solved this problem. है ना? But only problem with hydrogen right now is what? It's not economical. Unless you fulfill all these 4 criteria, no energy avenue, no fuel will be popular. वो popular होगा ही नहीं, है ना? Anyway, we'll move on to the next one. UPSC asked one question from biology recently. I mean, recent मतलब 5 साल के अंदर. And the question was regarding photosynthesis, right? Now you see that photosynthesis, और question क्या था? Which of the following statements is true regarding photosynthesis? Now anybody will read this question paper, jump out of the joy कि ये तो बना ही लेंगे, है ना? And the first statement was that it release carbon dioxide and it takes oxygen. बहुत लोग इस पे लगा के चले, जो उल्टा लिखा हुआ था बेसिकली, है ना. बिकॉज इट टेक्स कार्बन डाइऑक्साइड, रिलीज ऑक्सीजन. तो गैस वाला यही क्वेश्चन था. आउट ऑफ 4 ऑप्शंस देयर वाज ओनली 1 ऑप्शन दैट वाज इंडिकेटिव टुवर्ड्स कार्बन डाइऑक्साइड एंड ऑक्सीजन, एंड दैट वाज रिटन इन सच अ वे कि पढ़ने में आपको लगे कि अच्छा यही सही होगा. नॉर्मली क्या पढ़ते हैं कि इन प्रोसेस ऑफ फोटोसिंथेसिस प्लांट्स टेक अप कार्बन डाइऑक्साइड एंड रिलीज ऑक्सीजन. वहां पे क्या लिखा हुआ था? इट रिलीज कार्बन डाइऑक्साइड एंड टेक अप ऑक्सीजन. लाइक पहले कार्बन डाइऑक्साइड फिर ऑक्सीजन इन द सेम फैशन, बट वर्ड हैज बीन चेंज्ड, रिलीज एंड टेक अप. अच्छा, क्या होता है ना, क्वेश्चन को हम और आप जब पढ़ते हैं ना, तो वी डोंट फर्स्ट क्वेश्चन के साथ जो हमारी इंटरेक्शन होती है, इट्स नॉट वर्ड बाय वर्ड, इट्स लाइक फेस वैल्यू ऑफ द क्वेश्चन, है ना. तो उसके बाद 3 ऑप्शन जो थे उसमें क्या था? एनर्जी की बात की गई. पोटेंशियल एनर्जी, काइनेटिक एनर्जी, ये एनर्जी, वो एनर्जी. अब क्या होगा? फोटोसिंथेसिस एनर्जी, पीपल विल नॉट रिलेट विद दैट, मेजॉरिटी ऑफ देम. बट द राइट आंसर वाज इट कन्वर्ट्स फ्री एनर्जी इनटू पोटेंशियल एनर्जी. अब सोचो, it was a question of photosynthesis, but the right answer was, out of 4 options there is only one right answer, that it converts free energy into potential energy. Free energy means what? Sunlight energy is a free energy, and a starch बनता है. Starch is a chemical energy which is just stored. In energy which is just stored is known as what? Potential energy. You get my point? So these are, I mean, easy question, but it requires presence of mind, connectivity. Anyway, now nuclear agriculture, nano agriculture, we'll talk about agriculture nanotechnology. So again, with the help of flowchart, we'll try to understand it, because agriculture is very important. India के point of view से भी important होता है. So one is called crop improvement. One aspect is crop improvement. Right. Another one is crop protection. Crop protection. Or you can write crop nutrition. Three area we can talk about. And in normal agriculture also we talk about 3 area. I mean, normally if you go for the agriculture, what you will do? You will try to select the crop variety which can give you more output. Then you will protect your crop from any pest attack or any kind of infection. And then you will always try to provide better nutrition to the crop. तो यहाँ पे जो आएगा nanotech का area, that will be fulfilled by nanobiotech, nanobiotechnology. We'll talk about that just. This is called nanopesticides. Nanopesticide. And this will be done by nanofertilizers. Right. We don't have example of any of these two. I mean, experiments are going on, but now because of Indian, we have example over here that is for nano urea liquid. Nano urea liquid. But conceptual point that we can do all. Nanotechnology has a very close relationship with biotechnology, because nanotechnology has given us a kind of capacity and skill to enter inside the cell and manipulate the cell at nanoscale. And for that you need to understand just one thing, that whether it is animal cell or plant cell, animal cell and plant cell both are at micro scale. Which scale? Micro scale. So let's say cell, this is cell, and cell it at which scale? Micro scale. That is 10 to power minus 6 meter, right? And everything is happening inside the cell. सारे activity कहाँ चल रहे हैं? Cell के अंदर. So if we talk about inside the cell, inside the cell. So inside the cell किस पे आएगा? Which scale? Nano scale. We got 10 to power minus 9 meter. So that is one region that wherever the living contents are there, now we are able to reach to that living content by 2 ways. अभी तक जो बात की थी बायोटेक में लिविंग सेल्स के अंदर पहुंचकर मैनिपुलेट करना कैसे पॉसिबल हुआ था? विद एंजाइम्स, एज सिंपल एज दिस, विद बायोमोलिक्यूल्स, राइट? इधर यू डू आई मीन आरएनए यूज करते हो, डीएनए यूज करते हो, एंजाइम्स यूज करते हो, प्रोटीन्स यूज करते हो। तो जब तक आप आरएनए, डीएनए, एंजाइम्स, प्रोटीन यूज कर रहे हो, ये बायोटेक है कि नहीं? एंड द मोमेंट यू चेंज द वे दैट नाउ यू आर यूजिंग इंस्ट्रूमेंट, some lab grown chemicals, then it is what? Nanotech. It is nanotech. So nanotechnology has given us edge and capacity and skill to get inside the cell and manipulate the things directly over there, right? And that is how many scientists in India and in the world level also, they are able to manipulate the DNA with the help of nanotechnology. थाईलैंड में एक यूनिवर्सिटी है, चैंगमाई यूनिवर्सिटी, और वहां पे एक साइंटिस्ट ने पहली बार वर्ल्ड में फर्स्ट टाइम ऐसा हुआ था, दे चेंज द कलर ऑफ राइस बाय क्रिएटिंग अ नैनो होल इन द सेल ऑफ द राइस. So they created a nano hole in the cell of the rice, and through this nano hole they manipulated the DNA, and the rice color has been changed. Only color has been changed, nothing else. कुछ खास और भी नहीं. You get my point? A Kolkata based scientist had done the same thing with the flour. He changed the color of flour from white to purple, and with the help of nanotechnology. That means nanotechnology has a potential where DNA expression can be changed. अभी तक हम कलर वलर चेंज कर रहे हैं. मे बी इन फ्यूचर वी आर एबल टू मैनिपुलेट द न्यूट्रिशनल एस्पेक्ट, सम न्यू थिंग्स वुड कम अप, पेस्ट रेसिस्टेंट, कुछ भी कर सकते हैं. So that is about what? Nano biotech. You can write just one point. With the help of nanotechnology, intracellular mechanism can be manipulated. Can be manipulated. And it can lead to genetic changes. In crops. Genetic changes in crops. Right. That will be enough. Second is what? Nanopesticide, crop protection. Pesticide is a very bad thing for the agriculture, you know that. One of the reasons that we accepted, India opened from GM crop in 2002 when we started doing BT cotton, was to save environment. है ना? इंडिया में फर्स्ट टाइम जब हमने जीएम क्रॉप को एक्सेप्ट किया था, इट वाज नेबर प्रोडक्टिविटी के लिए. तो पहली बार हमने जीएम रबर वाज द फर्स्ट टू प्रोडक्टिविटी. वही है प्रोडक्टिविटी के लिए, डायरेक्ट प्रोडक्टिविटी के साथ वही लिंक हो रहा है. तो व्हाई डिड वी एक्सेप्ट this BT cotton? To save the environment, to protect the environment from massive use of chemical pesticide that was happening in cotton growing states, right? Because we know that pesticides are poison, right? Now the thing is that once you spray pesticide, then only a fractional part of pesticides will be used in killing the pest. More than half of the pesticide that will be spread all over. It will enter into the food chain to the crop. It will go to the water. It has reached to the water table also. It will kill the other part of biodiversity. It will pollute the air and soil, and ultimately it will come back to us. That is why with the मतलब ग्रीन रिवोल्यूशन हुआ, ठीक है, बहुत अच्छी बात है, लेकिन उसके हमने प्राइस भी पे किए, राइट? Overuse of chemical pesticide, overuse of chemical fertilizers. Haryana, the part of Haryana and part of states has become, barren land. खत्म हो गया, saline हो गया, because it was not scientifically used. People just had one thing: more use of fertilizer, more productivity. It's not like that. If you use fertilizers more than required, your soil will be gone. More use of pesticide, it is not going to kill the pest more. पेस्ट तो ऐसे ही मरेगा वो. नाउ इट इज गोइंग टू हार्म अस. तो अभी क्या कर रहे हैं ना, द सेम वे, लाइक टारगेटेड ड्रग डिलीवरी, याद है आपको? वी जस्ट टॉक्ड अबाउट. कैन वी थिंक अबाउट टारगेटेड पेस्टिसाइड डिलीवरी? सोचो. that pesticides are also locked in some nanomolecules, and it will be targeted to the pest because every pest are different, right? अगर अलग स्पीसीज है तो डीएनए अलग है, डीएनए अलग तो प्रोटीन अलग है. So now this pesticide will be targeted, it will be just unlocking itself to the target pest, leaving all the other part of biodiversity spared. And if it is not unlocked, it will be inert. अगर हमने खा भी लिया, पी भी लिया, तो हमारे बॉडी में शायद कोई एक्शन नहीं. अब यहाँ पे शायद वर्ड लगा हुआ है बिकॉज नैनो टेक्नोलॉजी में कुछ भी नहीं कह सकते अभी. बट यस, अप्रोच इज गुड. So you can write one more point. It will be based on pesticide. It will be based on targeted delivery system. Targeted delivery system where pesticides will kill only the harmful pests, without killing other part of biodiversity. Right. So that you will understand with that only. Now this is important, although UPSC has not asked any question. India is the first country to develop it. Right. You can write. Next one. Nano fertilizers can provide better nutrition to the growing crop. For example, growing crop. For example, India has developed world's first nano urea liquid. World's first nano urea liquid. Right. Next you can write. This has been developed by IFFCO, Indian Farmers Fertilizer Corporation. IFFCO. Indian Farmer Fertilizer Corporation. Next you can write. Nano urea liquid has following benefit. Although अभी थोड़ी controversy में है ये. Now there are one group who is saying that it's not as it has been promised. But ठीक है, government है अभी तक कुछ बोला नहीं. Nano urea liquid has following benefit. Following benefits. Has following benefits. First benefit. Right. Urea uptake or nitrogen uptake. Nitrogen लिखना. Nitrogen uptake of nano urea liquid. Nitrogen uptake of nano urea liquid is 80%. Which is double of normal urea. Now, what is this uptake? It's a plant uptake. Let's say you spread some urea like urea pearls. So, only 40% of nitrogen that urea can offer will be taken up by plant. So, you release urea will dissolve in water, nitrate will come up, and only 40% of nitrogen will be taken up by plant. 60% of the nitrogen will be there in the soil. It will go somewhere, it will do pollution and all kind of thing. But if you use nano urea liquid, the uptake of the plant will be 80%. Only 20% will be left behind. है ना? तो सोचो 40%, 80%. That means urea amount will be reduced to what? 50%. जितना हमें यूरिया चाहिए था इंडियन एग्रीकल्चर सिस्टम में, उससे घट के कितना हो जाएगा? 50%. बिकॉज अपटेक इज डबल. पहले हम 100 kg डालते थे, 40 kg यूज होता था. अभी हम 100 kg डाल रहे हैं, 80 kg यूज हो रहा है. ऐसा मान लो तो यूरिया इंपोर्ट कम हो जाएगा ना फिर. यू कैन राइट. It will decrease. It will decrease. Second point. The overall urea requirement by 50%. It will decrease the overall urea requirement by 50%. This will further decrease the urea import. Urea import. Next one. It will have minimum environmental impact. It will have minimum environmental impact and related pollution. And one more, the agricultural productivity will increase by 8%. 8. Agriculture productivity will increase by 8%. 8%. Right. So that is about this nanotech. One more thing that you should know in nanotechnology, that nanotech in India. Just one thing that you should know, because it's an easy one, that we are under nano science and technology mission, right? That we call it nano mission also. So already in India, nano mission is going on, and we are exploring all possible avenue of it. In this nano mission, we are focusing on few things, that is infrastructure development. Second one is HRD, human resource development, human capital development. Then inclusion of private company, promotion of R&D, and promotion of international collaboration. Now this is something that you need to understand. Private company must come into this. So इंफ्रास्ट्रक्चर डेवलपमेंट हमने किया हुआ है, बहुत सारे सेंटर ऑफ एक्सेलेंसेस हमने लॉन्च किए हैं. HRD भी ठीक ठाक है. Inclusion of private sector, private companies into this, promotion of research and development, promotion of international collaboration. Only thing we are lacking, that participation of private company into this sector is not as much as it should be, right? Why? Because one thing you should know, that nanotechnology is known as general purpose technology. General purpose technology are those technology that we are going to use extensively in our life. Moreover, nanotech is a material science. It's a part of material science and it's a part of general purpose technology. If you want to compare anything with nanotechnology, which is very closest to it in terms of impact on our own life, then it's a plastic and polymers. Plastic and polymers are again a part of material science and it's a general purpose technology. If you compare, you will understand. Because plastic and polymers are material science, you know that. And it's a part of general purpose technology because plastic को हम कैसे कैसे यूज करते हैं सोचो आप. In all sphere of life we are using it. Similarly, in future, nanotechnology is still, I mean, at the present time, nanotechnology comes under material science and nanotechnology is converting into general purpose technology. एनर्जी में भी यूज होगा, मेडिसिन में भी यूज होगा, क्लोथिंग हमारी होगी, कॉस्मेटिक्स, मेडिसिन, I mean, you name it, it's there. Rather, I'll tell you, it's already there. Although we don't know about it. हमें पता नहीं होता कि नैनोटेक हम यूज कर रहे हैं. अगर आप रिंकल फ्री शर्ट पैंट खरीदते हो, नैनोटेक है. अगर आपने चश्मे बनवा रखे जिसमें ब्लू कट है या स्क्रैच रेसिस्टेंट है, वो भी नैनोटेक है. You get my point? Many creams and many cosmetics are coming which are nanotechnology. Right? Nowadays, odor free and no wash fabrics are coming. It's a nanotechnology. If you go and buy some racket of tennis and badminton, it's very lightweight and very strong. It's nanotechnology. It's already there. It's already slowly coming in our life. Right? And it will come like the plastic has come in life slowly, and now we cannot think about our own life without plastic. Although we know that plastic is a menace now, लेकिन within 100 years ऐसा हुआ ना कि प्लास्टिक हमारे जीवन में एकदम घुस गया. Like in my class, I always say that you cannot imagine your life without plastic. कई बार ऐसा लगता है कि प्लास्टिक क्या ही है, छोड़ देंगे, और छोड़ के देख लो एक दिन, आप यहां से घर नहीं जा पाओगे रूम तक, क्योंकि जूते वूते निकालने पड़ जाएंगे ना. फिर तो बाहर में कोई अगर व्हीकल भी नहीं, तो इट्स वेरी डिफिकल्ट. कॉपी वॉपी फेंक दो वहीं पे, क्योंकि स्पाइरल बाइंडिंग भी प्लास्टिक की है, पेन प्लास्टिक का बना हुआ है. सो 1000 times more impact is coming when nanotechnology will mature. Because प्लास्टिक हम खा तो नहीं रहे, प्लास्टिक कैंसर सेल को तो नहीं ढूंढ रहा है कि नहीं, प्लास्टिक को फर्टिलाइजर के तौर पे तो यूज नहीं कर रहे हैं, लेकिन वो तो वहां भी करेंगे. इट विल बी इंटीग्रेटेड विद एग्रीकल्चर, हेल्थ, एनर्जी. राइट? सो नाउअडेज we are making clothing, we are making sports equipment, we are making war equipments, we are making energy avenues, we are having this nano urea liquids, pesticides, medicines and all. Right? That is why private company must come in. Right? That is one thing you should be aware of. सोचो प्लास्टिक को अगर गवर्नमेंट ही रेगुलेट करती तो क्या प्लास्टिक इतना वाइड स्प्रेड यूज में आता? ठीक है, ये बात बिल्कुल माननी चाहिए कि प्लास्टिक is a bad thing that happened to human being. We could have controlled plastic much better that we have done. क्योंकि 1930s, 35 और 40s में जब प्लास्टिक आया था इस वर्ल्ड में, then people were overwhelmed by the benefits of plastic. उस समय कहां सोचा था कि नाइलॉन will create some problem? तब तो ये लगा था कि नाइलॉन such a wonder material. क्योंकि उस समय तक क्या था ना कि पीपल हु आर वेरी रिच, दे वुड वेयर द ग्लव्स ऑफ व्हाट, सिल्क, व्हाइट सिल्क का ग्लव्स पहनते थे. दैट बेसिकली वेयरिंग अ ग्लव्स मींस व्हाट, दैट यू बिलोंग टू द अपर स्टेट ऑफ द सोसाइटी बिकॉज यू कैन अफोर्ड इट. सॉक्स पहनना उसमें बहुत बड़ी बात थी कि भाई सॉक्स, क्योंकि वो भी सिल्क के, और कितने चलते थे सिल्क के सॉक्स. अब आया नाइलॉन, जनरेशंस पहन सकता है उसको एक के बाद एक, फटेगा नहीं. And it look nice, it's so cheap, and it all over it become range कि अब everybody will wear this. तो उसमें तो बेनिफिट था कि बेनिफिट है. And then nylon, polyester, terylene and polypropylene and PVC and everything started coming in. And after a certain point we realized that we have done something very bad. In my lifetime I have seen in my village, if you go to the grocery shop, so they have newspaper. No matter what you are going to take, in newspaper they will keep it they will fold it, and there was jute, thread, and they will wrap it and then give it. There is no thali and nothing like that. And now all of a sudden, in my lifetime, I have seen that this thali has, this jute thread and this newspaper has gone, and polythene taken over. It has started in my life only. And now in my life also, I am seeing that now polythene is being banned. In just a time span of 30 years, I have seen all. I am 45, but 30 years के अंदर मैंने देख लिया ये सब कुछ. You get my point? So that kind of, because that time it was a rage. People would be kitna convenient. And that is one of the issue with the nanotechnology also. That is why we are holding back. One thing we know, that unless private company will come, this technology will not mature and go in the way that we want. क्योंकि अगर नैनो टेक्नोलॉजी से कपड़े बनने हैं तो कपड़े गवर्नमेंट तो नहीं बनाएगी ना. गवर्नमेंट तो कहेगी नैनोटेक से बुलेट प्रूफ जैकेट बनाएंगे, किसके लिए? आर्म्ड फोर्सेस के लिए. हेलमेट बनाएंगे, वॉर इक्विपमेंट बनाएंगे. बट स्पोर्ट्स इक्विपमेंट बनाना प्राइवेट कंपनी बनाएगी. बट एट द सेम टाइम वी आर गोइंग स्लो बिकॉज वी डोंट नो व्हाट काइंड ऑफ मैनेस इट इज गोइंग टू क्रिएट. राइट. अगर आप साइंटिस्ट के, आप होता क्या है ना कि व्हेनेवर द न्यू टेक्नोलॉजी इज कमिंग अप, देयर विल बी वन आर्टिकल विच विल बी इन फेवर ऑफ नैनो टेक्नोलॉजी. वो पढ़ के लगता है कि नैनो टेक्नोलॉजी आ जाए तो बहुत बढ़िया होगा. देयर वुड बी सम आर्टिकल विच विल वर्क एंड टॉक अगेंस्ट नैनो टेक्नोलॉजी. अगर उसको पढ़ लोगे तो लगेगा कि बस वर्ल्ड खत्म होने वाला है. बट देन इट्स अ बैलेंस. तो चलो, विल सी यू टुमारो एंड विल टेक अप द नेक्स्ट चैप्टर लाइक स्पेस टेक, IT एंड कम्युनिकेशन. If you have any doubt, you can ask.